अभी तक ज्वाइन नहीं किया किसने? ये घर है सब भ्रमण कराओ ऋषभ भ्रमण कराओ
हाँ यही है,
यही है
हाँ हाँ
पक्का ऋषभ ठीक है
हाँ यार मेरे को पता नहीं। बोलो क्या नाम है?
यहाँ पे है ऋषभ
HRU HU
SHEP Webinar starting. Please wait. Submission.
Okay. Honorable Professor Kiran Kalia, Director NIPER-Ahmedabad, eminent speakers, Professor Anand Parakar, Director and Professor, School of Pharmacy, University of Blackford, Professor Nizar Al-Shari, Faculty, Jordan University of Science and Technology, Mr. Arun Mishra, Head, Global Regulatory Affairs, Unilever India, my dear colleagues and dear participants, a very warm welcome and good afternoon to all of you. It's indeed a very beautiful day when we are witnessing an active and united participation from various parts of the world, including the participations from various countries, including USA, UK, Jordan, India, Israel, and many more. On behalf of NIPER-Ahmedabad and organizing committee, I, Dr. Rakesh Tekade, welcome you all for this webinar that entitles Future with Interdisciplinary Research Collaborations. Friends, if we quickly try to Google down the meaning of interdisciplinary research, probably we all get that interdisciplinary research is a technique in research where fields of different science and technologies and disciplines come together unitedly to resolve a given problem. I hope that you all will agree with me, if not all, interdisciplinary research is having the capability to resolve many of the problems of this decade, including the ongoing COVID-19 pandemic. Still, friends, there are several quandaries in our mind, especially the young minds. That includes questions like, what is the practical meaning of interdisciplinary research? Why is interdisciplinary research important? Is it important or not? What are some of the real life examples of interdisciplinary research? And there are like many more quandaries that goes on in our mind. And this is where our webinar is exactly focusing on. So friends, before we start our today's webinar program, I request all of you to please stand up and join us for the national anthem. Friends, before we start,
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उज्जल जल जनगणना अधिनायक शरणा में जागे शुभ आशीष मांगे गाहे तब जय गाथा जनगण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे शिव जय 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 हे
thank you very much. Thank you. Thank you for making the environment and the arena so pleasant with this national anthem. To go ahead, I introduce Professor Kiran Kalia. Professor Kiran Kalia, Director NIPER-Ahmedabad, is indeed main pillar and the SOP of interdisciplinary research work at NIPER-Ahmedabad. She had worked tirelessly to see where the NIPER-Ahmedabad is currently standing today. I invite and request Honorable Professor Kiran Kalia, Director NIPER-Ahmedabad, to please come on the stage and inaugurate the sessions and give us her blessing for a commendable webinar. I request all of you to please give a huge round of applause for Dr. Kiran Kalia.
Thank you, Dr. Rakesh. It's indeed a very pleasuresome time for me to inaugurate this webinar. On behalf of NIPER-Ahmedabad family, I feel privileged to welcome all the speaker participants and colleagues for taking part in this webinar. This webinar is certainly touching a very valid subject. Of that, it is interdisciplinary collaborative research. To talk on the same and share the views, today we have Professor Anand Paratkar from University of Bradford, Dr. Nizar Al-Shari from Jordan University of Science and Technology, and Mr. Arun Mishra, who is heading the Global Regulatory Affairs at Unilever India. I, on behalf of NIPER-A family, once again join my organizing committee to welcome you all to this webinar. And I hope that this webinar will be beneficial to all of you scientifically. There is a popular quote to say that if we move together unitedly, then success takes care of itself. The motto of this webinar is to bring together the academicians, the scientists, and the industry regulators to strengthen the translational aspect of our research. And thus, thereby, we realize the value of interdisciplinary research. Today, this platform is witnessing the attendees from several countries, including India, US, UK, Israel, Jordan, Malaysia, to name the few countries. It includes bachelor, master's, PhD, postdoc, faculties, as well as the industry professionals among us to attend this webinar. I welcome all of you wholeheartedly for this webinar and also invite you to visit the NIPER-Ahmedabad personally whenever you all get a chance to do so after the ongoing pandemic time. With this, I declare the session open for the talk by the eminent speakers. And once again, I thank you all for your participation and best wishes to all of you for a great, successful and productivity of this event. Thank you very much.
Thank you, Prof. Kiran, for giving such an inspiring speech and showing us the path forward. A special thanks to Professor Kiran for giving a big deadline of today. I must say a very big deadline for the today, especially for the young minds attending the webinar with us today. The tagline given by Professor Kiran is, if we move together unitedly, then success takes care of itself. Indeed true, 200%. Thank you very much, Prof. Kiran. So before beginning the scientific sessions of today's webinar, we would like to give a brief introduction about the research and teaching activities of NIPER-Ahmedabad through a short video. I request all of you to please have a look at it.
National Institute of Pharmaceutical Education and Research, Ahmedabad, a commitment to world health, and an institute of national importance. Started in 2007 and presently offers master's and PhD program in biotechnology, natural products, pharmaceutics, pharmaceutical analysis, pharmacology and toxicology, medicinal chemistry, and medical devices. From August 2016, the institute is functioning at its own campus at Gandhinagar with the state-of-the-art research facilities. The founding director, Professor Kiran Kalia, is the torchbearer of NIPER-Ahmedabad and is leading the institute to make it one of the top premier institutes across the globe. By her relentless efforts, the institute has secured impressive NIRF ranking and catapulted to topmost pharmacy institutions of India in the category of teaching, learning, and resources. A strong administrative team ensures the smooth functioning of the institute. NIPER-Ahmedabad has numerous high-impact publications, patents, and awards to its credit. It has built strong international collaborations and achieved 100% placements. Institute has well-equipped classrooms with all the modern tools of teaching and research. It became the first institute of India having electronic lab notebook for capturing all research work with highest data integrity and help country by providing trained human resources to pharma industry. Many distinguished guests have witnessed the progress of NIPER-Ahmedabad. Institute is equipped with advanced instruments like NMR, FSM, LCMS, QTOF, flash chromatography, gel permeation chromatography, porocytometer, microplate reader, freeze dryer, DSC, TGA, ultracentrifuge, polarimeter, hypoxia chamber, and gas chromatography, confocal microscopy, and small animal imaging facility to carry out quality research. Well-equipped classrooms with all the modern tools for teaching are its other assets. The Drug Development Lab is working in the field of formulation, complex analytical evaluation strategies, and medical device development. Drug development researchers are working on formulation development for NCEs and generic molecules to fulfill countries' needs. They are extensively involved in the development of controlled and site-specific delivery of therapeutics in cancer, diabetes, and neurological disorders. The group is also engaged with complex analytical characterization, reverse engineering, pharmacokinetic, toxicokinetic, and metabolic profiling. They also perform impurity profiling, drug excipient compatibility, and stability profile of pharmaceuticals with state-of-the-art analytical facilities. The lab is also focusing on development of smart biomaterials, 3D tumor microenvironment, 3D bioprinting, porous materials, and engineered strategies for ocular, cardiovascular, and orthopedic applications. The Chemical Biology Lab explores the strategies to discover novel scaffolds from terrestrial and marine resources and developing lead-based design of natural scaffolds against diabetes and cancer. The lab is also exploring various strategies like CH activation, green chemistry, and other synthetic organic methodologies to synthesize novel neuroprotective and anti-cancer agents. The quest of clinical research is supported by proteomics, genomics, and biomarkers for cancer, diabetes, and its microvascular complications. The Pharmacology Lab emphasizes on stroke research involving therapeutic implication of stem cell delivery in small and large animal models for ischemic stroke, and the lab is identifying novel therapies for Parkinson's disease and cancer. NIPER promotes collaboration, innovation, and translation. The national and academic integration is achieved through celebration of national festivals, conducting international conferences, and industrially relevant workshops. Together, we make the difference.
Thank you very much, Ashish. With this, let us start our today's scientific session. To start with, we have our first speaker, Professor Anand Parakar. Professor Anand Parakar is a personality which do not need an introduction, especially when you talk about pharmacy. But still, to introduce him, he is an academician turned entrepreneur, an industry consultant, book writer, group leader, and the list going on. He is currently serving as a full professor in the School of Pharmacy at University of Bradford, UK. He is the director of the Center for Pharmaceutical Engineering Science and interdisciplinary chair in pharmaceutical engineering science. He has published several number of research papers in high impact journals. He has been awarded with several prestigious awards, including EPSRC funding. He is a recipient of JSPS International Joint Project, recipient of UKRI Fellowship, Yorkshire Entrepreneur Fellowship, recipient of Royal Science Network Award, as well as Business Innovation Award. If we try to Google down about Professor Anand, then probably you will get endless number of information there. But still, there are some recent accolades about Professor Anand Paradkar. He has been recently awarded as the best employee of University of Bradford. He is serving as an active editorial board member of various international journals. And still, with this very short introduction about Professor Anand, I request Professor Anand to please take on the lead and the stage. Please welcome Professor Anand with a huge round of applause. Professor Anand.
Good morning, everybody. Good afternoon, and very warm thanks from my side to NIPER. This is my first time I had any interaction opportunity to interact with NIPER-Ahmedabad. And to look at when I saw the activities, it is great. It is really something which we are proud of, NIPER team. First of all, thank you from Professor Kalia and Rakesh to invite me for this, giving me opportunity on this platform with Dr. Mishra and Dr. Nizar. I would like to share some of our work which we do at Bradford at Center for Pharmaceutical Engineering Science. Mainly, it is an interdisciplinary center. So I would like to start my presentation. Just give me a minute. Today, really, I changed my internet supplier. So there was some yesterday midnight it had changed. So it has given some problem me in connecting with in the morning. Apologies for that. Whether my screen is now shared?
Yes, Professor.
Okay, thank you. So the topic which we have selected today, Rakesh and me, Rakesh suggested, and I also think that this is a good topic, which is interdisciplinary research for innovation and strong out innovation. So to start with, I will just give you brief background about idea about Bradford, University of Bradford. So this is our university at very small university at Bradford, which is around 200 miles north of London. So whenever you are in the UK, please do visit University of Bradford. So today, luckily, same weather is it is rare to find such Bradford, and today also have similar weather, which is a good thing. So without taking much time, I will give you some idea about the pharmaceutics activities at Bradford. In 2005, I came to Bradford as on a 3 months visit on Royal Society Fellowship, work with Professor Peter York. Professor Peter York, that time, had successfully completed or commercialized one technology, which was supercritical fluid technology. And what in 1994, really, he and his PhD student developed the technology for precipitation of particles from supercritical fluid, which were superior or excel for inhalation purpose. So company spin out in the backyard of the university, which was Bradford Particle Design. And in 2001, it was sold to therapeutics for $220 million. That was one of the best success for the university. So here is Peter York. This is Hannah, his PhD student in the company, and Green Humphrey. But so here you can see print was benefited. The innovation at Bradford is really, we have very strong heritage, and I was lucky to work with Peter York. So with this, university also was benefited. University funded this building, established this center. That time it was Institute of Pharmaceutical Innovation. So I joined in 2005. I came 7. I came again UK postdoctoral fellow, and university was very kind to appoint me as professor in 2008. In 2010, so in 2008 when I came, really it was very bad period for from funding point of view. Economy was down. So university has restructured everything. Its centers, funding, distribution, and university decided to form a Center for Pharmaceutical Engineering Science, and I was given opportunity to lead it. So for last 10 years, I'm director for Center for Pharmaceutical Engineering. So what is Center for Pharmaceutical Engineering Science? Main thing, it is an interdisciplinary research center. So this is the building where all our pharmaceutics mainly research resides, and across the road another building where engineering research resides. I started working across these two centers. We have excellent capability in no doubt pharmacy, solid state, mainly material science, and so chemist, pharmacist, material science people. So they work on structure and properties of the material. From pharmaceutical point, we performance, and from engineering expertise comes about the processing. We have one of the best hot melt extrude or melt processing center in the Europe, across the Europe. Good micro molding, injection molding, all sorts of capabilities. And we are completely translational. We do blue sky research, but at the end, exactly like NIPER, our objective is how companies can be benefited, how economic will be benefited, how society can be benefited. So translation is the main part of our research. So we work with companies starting from pre-formulation level to process development, but many times we get process analytics nowadays, because big companies are more into process analytics, QBD type things, formulation development, product development. So pre-formulation, formulation, processing, process development, and process analytics. But apart from that, the main important thing which we started is we do some commercial or our own patents. We try to commercialize on our own or in collaboration with the companies. It is similar to what you do at NIPER via or ICT via BIDAC projects. So the first thing which attracted me to Bradford, and what we realized and what we do have advantage for me, because it is a small university. We have very strong STEM faculty. So we have excellent engineering, chemistry, biology, and pharmacy. So generally, when we tried to do any creative thinking process, idea generation process, we pharmaceutics group or pharmacy, it is one of the major limitation for generating ideas. So what we have decided is to have interdisciplinary brainstorming sessions, which may be a very small group. It may be it is generally investigator or research process we adopt, and that really helps significantly to generate new ideas. Especially, for example, from pharmacy point, I can think something, but I do not know about engineering processing, where my engineering colleagues are working. So as we know, there are incremental innovations which are generally occurring in the companies where generic forms are there, or a new product of existing service. So only you are increasing the market, but degree of innovation is limited. Whereas breakthrough innovations, like I saw just now, you are generating some new materials for medical devices, or some new crystal forms, and especially I saw it is at NIPER, it is very strong chemistry also, natural sciences and chemistry. So most of the innovations occurring at NIPER will be breakthrough innovations, and there are some radical innovations where the product, we develop a product for totally non-existing services. And best example which has come into picture now is because of COVID, completely new type of business models are coming up, new products are coming up, like COVID vaccine. But as an academic, what we find as an early career researcher, especially my focus is on, I do like to discuss this more and more, because I get chance to interact more with early career researchers, and early career researchers do not have a network and chain available. And this is something which I try to do at Bradford, that how I can nurture the early career researchers up to the so what happens is we do something in the lab, but how it will translate to the company level, to the commercial level, and that it requires huge number of steps, and most of the innovations really fail because of lack of support, or basically I will say now the science or the stream which is coming up, which is improper or inefficient innovation management, is the major reason why innovations fail. This we identified, and we tried to set up a link for early career researchers, and even for me. I am at the same, though I'm now old, but not young. Early career is the same problems. So in the university, we have a structure at the center. What we do is we have innovations research and innovation support unit, which comprises of commercial patent person, commercial manager, business development managers, and legal team, who takes up, who will be conducting major part of commercial activities. But the ideas start at the academic level. So we have this is just few I have put here, but there are many of my colleagues who are involved. So mainly with whom I maximum work is my engineering colleague, who is Adrian Kelly, and there are 5-6 major engineering professors with whom as per project I work. Christoph Paluch is pharmacist, Venu is crystal engineer, Mohammed is mainly working on pharmaceutics, but protein type molecules, and Colin Seaton, who is my chemistry or mainly crystal engineering expert. And a group of early career researchers, their job is to deliver the project, to work on the innovations, to take and add to the creative thinking. So I'm very proud that in last 10 years we have really many others, but 4 innovations which we are currently working and taking to the next at different stages they are. So this is FastAct, which is a poor technology for for poorly soluble materials. Self-gel is for some medical devices. This is for ARS, abuse-resistant system, ARCAGE, a cage for efficient or use very innovative abuse-resistant device, and the most important, which is commercially at relatively further stage, which is effervescent product. And I will give you today idea about effervescent product. This graph shows this is generally what happens, and all of you must be, and you are better in a better position than me because you work with lot many companies and with lot of ideas. So we conduct research, we have invest lot of resources, your time and funding in research, but at the end it fails because in this we struggle in the valley of death because we do not have this bridge or the support, which is most important until we go for commercial success. For that, we try, like you get in BIRAC or DST fundings or company funding, we here major support from Innovate UK, which also allows us on many projects to collaborate with other countries, especially nowadays with India. And there are translational grants in the region. For example, to give, so Ahmedabad Corporation is funding some projects, or Gujarat government funding projects. Similar way, here region fields, for example, Leeds City region funds us some money. Till now we used to get from European Commission. Now I don't think we will be lucky to have that funding. One of my most successful way of working like BIRAC is knowledge transfer partnership, where we identify the gap between what is exactly technology or product requirement in the company which they cannot do, and academia can fill up that gap, can bring that expertise. So we develop the we have the expertise. We recruit early career researcher. Here is, I think, all of you many of you may know Shankar Katikhai. He's worked, Shankar and Bhagyesh, both of them worked at NIPER-Ahmedabad. So Shankar joined me as a knowledge transfer partnership knowledge transfer partnership associate, and he has taken one technology from the university and transferred it to the company. So this is a excellent way to generate innovators as well as the managers for tomorrow. I won't go much more. I will explain in between about the processes and how this is, everything is interdisciplinary. Only you need disciplines like management, you need discipline, social sciences, everything. Initially we used, when I came here, I thought only pharmacy and engineering, and now government funding agencies have also realized that it is to translate innovation, you need to have more than only sciences, regular conventional sciences. We need social science contributing to us. We need management sciences contributing to us. So I'm going to give you a simple example and how multidisciplinary efforts worked in this. So this is an example of simple effervescent formulation, which all of us have done at undergraduate level in dispensing, pharmacy, and other places. That time we didn't realize that what is problem with if there is any problem with the formulation. When I went to the company and realized that one of the major problem in effervescent formulation is its manufacturing. What it generally contains, it contains citric acid and sodium bicarbonate. But citric acid and sodium bicarbonate in very small percentage of moisture, humidity, react to form effervescence, generates further water molecules which catalyze the further reaction. So this problem makes manufacturing of effervescent products very challenging. And to address that, it's the only solution what companies generally do is they dehumidify the manufacturing area, packaging area to around 20% or 10% relative humidity. Imagine manufacturing under 10% humidity or 20% humidity is highly expensive. It is very expensive. It has for the workers working in extremely dry atmosphere, product, your granules are over dried under that condition. So compression is difficult. You need more pressure. There is more wear and tear of your compression equipment. There are number of problems which you will face when you are working with this. So we try to understand what exactly is going on here. So what we realize that citric acid, so this is dynamic vapor absorption forms, isotherms of citric acid and sodium bicarbonate. So here you can see that citric acid up to 50% RH. So around 45 to 50% RH, it takes very small moisture. Here you cannot see it. And suddenly at 80, it takes large amount of moisture. Sodium bicarbonate, the relative is almost no moisture absorption. But when you keep these two together, the reaction takes place at even 30% relative humidity. That I will show you in the next graph. But so the challenge is instability of effervescent formulation during manufacturing, which makes your air hydrothermal conditions in the very challenging, and it is at least 3 to 4 times expensive. One thing which from Indian point of view, or Malaysia, humidity, high temperature areas, it is extremely difficult. It is far more expensive. So initially I was thinking, so if you see manufacturing in India of effervescent products is extremely expensive because you have high temperature. When you try to bring down temperature, humidity increases, bring down humidity, it is far 3 times expensive than here. And then you need packagings which are expensive packaging or non-recyclable. So for example, generally vitamins, supplements, we are using these type of plastic bottles with silica gel in the top, making it non-recyclable. So significant issues in the product. So what we thought, can we use crystal engineering to solve this problem of hygroscopicity or destabilization of materials? And at Bradford, we work on different forms of solids. We work mainly on amorphous, polymorphs, and co-crystals. Not much on salts, but these three are far more. So what are co-crystals? Co-crystals are, most of you know co-crystals. So co-crystals are crystalline materials where you have a drug molecule. Consider this is drug crystal, this is co-former. But in a co-crystal, that more than one component type of molecules are sitting, but the bonding between them is non-covalent bonding. What is advantage of this? The main advantage of this is dissolution enhancement, because the energy of the system changes. You are including some polar molecules also. So the molecule which has been filed as or approved by US FDA, that is valsartan sacubitril, but it is a salt also, and it is hydrate also. It is not only co-crystal. So somebody, scientists argue that it is co-crystal, this is a salt, but leave it. That is not our main objective. But these type of materials can be, our most maximum patents are for enhancement of dissolution. Bill Jones group at Cambridge, he has contributed so Professor Desiraju's group and Ashwini Nangia in India, these are pioneers or major contributors, mainly Desiraju's, and Professor Bill Jones in the UK, he has shown that how crystal structure, making paracetamol to paracetamol co-crystal, gives you paracetamol co-crystal has a layered structure, and tableting can be improved. Stability enhancement. This is the most important paper, which very simple paper, but this really made me think about something. So this is caffeine. So caffeine forms hydrate. And what Bill Jones showed that if you make oxalic acid co-crystal, so where the sites at which water molecule binds, replace it by oxalic acid, and now caffeine oxalic acid co-crystal will not be will not be hygroscopic in nature, or will not form a hydrate, not hygroscopic. And on the basis of this, I thought, can we do similar thing with the citric acid thing? So this is just basic principle was observed just in this literature. And on the basis of that, we generated the hypothesis that yes, if we generate, convert citric acid into co-crystal, it may it may become non-hygroscopic, and it will solve the problem. So this is technology readiness level, and this we have to define every time when we go to any funding agencies, especially translational. They ask at what TRL you are. So this was TRL 1. I had a concept, and then I prepared citric acid nicotinamide co-crystal, and what we observed, it absolutely does not take in moisture. So okay, we have proven that moisture uptake of citric acid can be controlled by addition of nicotinamide, making it not addition, converting it into co-crystal or new crystal form. Then we check whether really will it work with effervescent formulation. So this is your standard effervescent formulation. Just mix citric acid and sodium bicarbonate and keep it in DVS. You will see at a small amount of moisture is taken at around 40% RH, and suddenly what happens is there is loss of weight, which is due to loss of carbon dioxide. So whereas when we prepared co-crystal effervescent product with co-crystal, absolutely there was no weight gain, weight loss till 50 to 60%, and which is we want to process anything at 50% GMP condition. So we have proven our technology. Just concept was proven, not technology. Concept was proven. So we from TRL 1 to TRL 2. Here is we prepared some tablets, carried out stability testing, and this completely shows that experimentally at experimental level we have some proof of concept data. Here I can go to go and disclose it, not disclose really, start discussing with funders, investors, or and other mainly other experts in this area. So the next, before file a patent or and this everybody every one of us does, competitor analysis, patent analysis, and mainly, is there any market? I'm doing all these things. How big is the market? Because we have to invest resources from the university, mainly for me from the center. Is it worth? So we do proof. Also, one of my PhD student, Azad, now he is postdoc, but Azad did proof of market study. He was supported by the funding agency. They gave him 30,000 pounds, 35,000 pounds, gave him opportunity to visit all over the world to the trade shows, labs or companies, visit them and find out whether really this is a good opportunity. If it is a good opportunity, we are thinking about nutraceutical. Is there anything else on nuclear pharma? What other opportunities are there? So Azad did that study, which was excellent for his development and good for me getting work done sitting in the office. So what about that there are literature search and market survey showed that there are two major technologies. One is soda and second is topogranulation. So what is FR soda? All these technologies are barrier. They bring barrier between citric acid and sodium bicarbonate. So in case of FR soda, the barrier layer is formed on the surface of sodium where sodium sesquicarbonate layer is formed, and that sodium sesquicarbonate is less reactive with citric acid, and it delays or slightly reduces the rate of reaction. Where topogranulation has topogranulation is exactly how we used to carry out dispensing pharmacy practical, slightly hit the system, and water of crystallization from citric acid was released. Some reaction will take place, and a layer of sodium citrate is formed on the surface of. So this is controlled reaction. Effervescent reaction is carried out under vacuum. Moisture is removed, and a small, thin coat of sodium citrate is formed here, and reaction is slightly reduced. But you do both these, they are not efficient in controlling the reaction. You will not be able to carry out your process of 40 or 50. Still dehumidification is required, but it will retain more CO2 compared to the conventional one. Whereas in our case, in case of FR shield, it is a molecular level barrier, so simple, and all these things are very simple. So I will explain you later how many about processing step goes. So here the co-crystals are formed, and now these co-crystals are protecting reaction. It's a huge market. It's a huge market. Trends are changing, and effervescent products are still untapped market because of the processing challenges, storage challenges. So based on this, we filed patent in 2015, and in 2019 it was granted. Still, I'm waiting for patent in the other areas, other regions. Then the next part, what we did, and here comes really, this is the most critical part, that we sit down and take the business model canvas. If I had to take this technology, okay, I have invested now in the patent. Now how quickly what is my exit strategy? Who are the key partners? And here comes all the collaborators. I need to conduct now scale-up studies. I need engineering calculations. I need to understand exactly what is happening, science part. So chemists are required. So identify your collaborators, as well as the collaborators are not only academic collaborator, they are industrial collaborator. There is industrial research which has to be carried out. And one part which we generally neglect is industrial research. Industrial research is far more, is one of the critical part. And then what are the key activities? What is my value proposition? What I'm going to really achieving? How will I face? And all these things are good for me as well as for the early career researchers to streamline the process. Here, the commercialization team, and we calling external experts, and they help us in getting the things. So once that is done, we try to demonstrate it at laboratory level. So this is these are the tablets. So this is conventional tablet. In 12 hours, if you put it open, it will collapse. Whereas a FR shield, after 30 days under similar conditions still retains maximum CO2, is still stable in structure. And this we carried out under for GMP condition and at the company site. So we reached TRL 5, and unless we reach TRL 4, 5, funding, translational funding becomes difficult, because government is not very confident at up to TRL 1, 2, 3, generally EPSRC type funding. So fundamental science, you study first and then come to the, but in our case, we moved ourselves up to TRL 5. Still, I didn't know about the fundamental science of that to the great extent. And we started immediately, because main thing for the university to keep investing money is very challenging. Resources are limited, and sometimes, and most of the patents, all of us know that the commercialization for many patents is challenging. My first patent, when since I came here in 2010, I filed, and I lost 100,000 pounds. So 1 crore on that. I could not get the patent, and I could not translate it to commercialize it. But I learned from that. But that type of cost university had to bear. So university was after me that get it out quickly, then we'll invest on next one. So we try to, the university system helps with CDAs, MTAs, legal contracts, and how to identify industry, the terms of licensing, all those parts were done by the university, and we collaborated and transferred patent to the company. Really, that when we did that, the patent was not granted. So we were at TRL 6, but university and was under heavy pressure, and from center also I had to put money. So I have to put money. I was also under pressure, so we and after that, in 3 months, patent was granted. It's okay. So we started with a simple grinding, like what most of us do in the lab. So you can form co-crystals in a water and pestle by neat grinding, nothing else. Just take the two components or add some small amount of solvent and do it. In laboratory scale, we can do at 200 grams we did, and now commercial scale we are reaching to 400 kilograms. So in summary, what exactly we achieved through this? So it is an innovation, material innovation, citric acid made into co-crystal, and now it is non-hygroscopic, granular, free-flowing material. It also main achievement is manufacturing innovation. Now you don't need humidity, hydrothermal controls. Carbon footprint reduced significantly. And the next is main important thing is packaging innovation. Now you don't need plastic tubes and resistant packaging. You can have simple packaging for the material. So one innovation led to multiple innovations on the way. We have TRL 7, and now it is with the company. No doubt I do work with the company in collaboration with the company to scale it up, and I hope by early next year, product should be on the market. Then after doing this, and all of us know that patenting or IP protection delays publication, outputs are delayed, students suffer definitely because of this. But at the same time, if students are getting IP, is one of the best thing. And I'm going to discuss about outputs. This is one thing which now I'm trying to share with everybody. So then I went to Colin Seaton, my chemistry crystal engineering expert, and I asked Colin that what exactly is happening. I'm thinking that co-crystals are not hygroscopic. He said no. So he did modeling on different surfaces of with only citric acid on different phases of citric acid crystal. He put the water molecule and tried to see. and he did it for water molecule on the nicotinamide co-crystal also. The main he's suggested is it is not really hygroscopicity is decreasing because the material is the groups which are hygroscopic are sitting inside and the surface is becoming they are less exposed to the surface. But again, I don't need to be expert on modeling, so you need a strong collaborator. This is the most important thing, the collaborator with whom you can rely on two things. One is expertise and second thing is delivery. Most of the times collaborations fail because of lack of delivery or lack of trust between the partners. After doing this, I thought I have done excellent science and now I'm going to get it in very high impact factor journal. And this year I should confess, I learned many things. I focused on two publications, which I thought that this is excellent work which we have done and I need high impact factor publications. So I started from environmental chemistry and then it was rejected at maybe 3-4 journals. At the end, I published it in Crystal Growth Design. And Crystal Growth Design is not a bad journal. I really like this journal. But the problem was, I was thinking, when will I get environmental chemistry? When will I get high impact factor, a chemistry journal? And really, I was frustrated. And at that time, I realized there will be many people like me who will be getting frustrated. And I was really thinking, what should be done? What exactly is wrong with this? And I came across San Francisco DORA declarative research assessment. And the first thing which we have to remember that impact factor is not the assessment of, does not assess quality of your research. Impact factor was designed for librarians to purchase which journals they should purchase. On that basis, they can, there are a lot of variations. Publishing patent is also a type of publication. And why this everything has come up? Because all of us, all our we researchers are under heavy pressure publishing in high impact journals. And DORA, I'm not going to go through all these things, but do not come under pressure of impact factor. Be confident of your research, publish in good journals. And that doesn't mean that we shouldn't try for best journals. We should try our best, but failure to get it in the high impact journals is not, does not in anywhere mean that your science is bad. And then the same thing was mainly on this effervescent product. We received really prestigious BioNow award last year. And the other technologies are in different at different stages. So all this is basically work which has been carried out. I get more credit because of being a director, but my colleagues and the interdisciplinary team around me works for that. So thank you, and please, whenever you are, and I'm extremely thankful for the audience from all over the world. This is really good opportunity for me to share with you, all of you, some of my very small experience in terms of innovation. But innovation is something which we should keep on sharing, and it inspires others. Okay, thank you. Thanks a lot.
Thank you, Professor. It was certainly a wonderful talk, Professor Anand. Thank you very much. Thank you very much for enlightening us. And now the floor is open for questions. We have already received some questions. Since you have touched a very valid point on the output, especially from the PhDs. So I have one personal question, Professor Anand. It is obvious that most of the universities across the globe, they are making some publications compulsory out of some PhDs. So they have a mandate that you have to get at least say 1 or 2 or 3 publication out of your PhD to make you eligible for PhD submission. So being such an expert who might have been as an external examiner for many PhD theses, may I please ask your your words on the same or your view on it, how you look at it?
Yeah. So this is very interesting question because at Bradford we do not have any compulsion. In most of the UK universities there is no compulsion for number of publications because we need to identify, understand. For example, this is work from Sudhir. Sudhir could get publications, but there are others where really I have to stop their publications for commercial purpose. Now, should that student suffer? No. We even stop publication of thesis. We embargo thesis for 3 years, 5 years, going on online, depending on the commercial sensitivity of that. But said so, it is, we need to, what is basic point in making student to write a paper is to provide him excellent, to demonstrate that he has excellent writing skills, written communication skills, and his work gets peer reviewed. But if I am confident, an examiner is confident, it should not be the requirement. It should be, we should encourage, we should motivate, but to make students suffer from, for example, I will say last year, one publication which I tried, it has taken almost 8 to 9 months for me to get it even to reach to Priscilla. Otherwise, I would have stopped. So in other words, I would have stopped the student from submitting his PhD for 8 months because the publication is getting, but I was confident that work is good. It is, and after that, what I, at the end of, and this is part of my performance, tomorrow I have my performance event where I learned, what I learned is what the time which I invested on getting into high impact factor journal, is it worth? Instead of that, I would have done something, and written communication skills of the student were same. Apart from frustration, it did not contribute me anything. But when I am saying that, it has, I learned many things. What is required in the higher impact? Submit it? No, I know where to submit. I need to do some cost to benefit ratio type things, and I will submit to the appropriate journal. This DORA guidelines have also come because of this. We put lot of pressure on our early career researchers. You will not get promotion. You will not get to this point because of limited publications or low impact. In many countries, there are total number of impact factor or average impact factor. They calculate what sum of impact factors. It puts a lot of pressure on early career researchers. Rather than doing science, they go on impact factor, which is not the correct thing to do. And that is why you can see here, really journalist impact factor based should be eliminated. And this is going to come. This is tomorrow. In next 5 years, the impact factor based metrics will go and has to go, because we cannot make. So there was one suicide in US or Australia, I don't remember now, which really triggered the question, why this, we made this, brought this person under pressure. This lecturer, early lecturer, he was struggling. For example, like if I cannot publish till now in 10 impact factor journal, what is, so either I'm wrong, I'm not a good researcher, or something is wrong and we cannot keep on putting pressure on students, postdocs, and researchers only based on impact factor. Rather, we have to encourage them for better publications, but not better science, rather than forcing some bureaucratic impact factor system, as well as numbers.
Very well said, sir. So, as you said, yes, I agree with you that all of us must work and walk behind the skill development rather than walking behind the success. Very nice. Sir, now we got some interesting questions via various platforms from the audience. May I please narrate you, sir, very quickly?
Yeah.
All right. So, sir, our first question is, how can we separate incompletely formed co-crystal?
How can we separate?
Incompletely formed co-crystals.
A very nice question, very interesting question. He has identified the limitation of. So, incompletely formed co-crystals cannot be separated by in the solution state as well, and this is one of the challenge. So, when I didn't present the data on optimization of co-crystals, so this is very superficial presentation. Please refer to some of my publications on melt extrusion, co-crystallization also. So you can prepare co-crystals by solution state or solid state. The major limitation in solid state processing is impurities cannot be separated. Even not only components is also an impurity, cannot be separated by this. So people are presenting that there will be some synchrotron type things, but these are not practical at so you need to just the specs will have 89%. So for example, here we need to find out what is the maximum unreacted component I can allow in this product, so that still the activity will be affected. But generally, most of the product is about 89%, but you cannot separate. You cannot separate impurity from the even in the solution state. What you need to do is you need to design a method to convert it maximum, optimize your method, but you cannot separate it, or especially in solid state process.
To speak about molecular dynamic simulations down the line today during our today's webinar. So I got a very quick question about it. So can we apply these molecular dynamic simulations for the designing of co-crystals?
Oh yes, there are number of. So I'm not expert on this. But our most of the work nowadays involve some molecular simulations are required, because understanding about the crystal structure is involves that, and it for co-crystal design, screening everywhere, which will be the most stable form stability assessment also. We take help from Colin means crystal engineers and modelers.
Here in our chat box, he is asking one question: can we use ultrasonic crystallization technique to generate co-crystals?
Yes, we can. And rather, we have there is a company, Prosonics. Okay, so if you are interested, go and see Prosonics website. Prosonics did this, but at the lab scale, I did it, and there are a couple of 3-4 publications. This part.
So, sir, one of our student is asking the question: what is the rationale followed for the selection of co-former?
our first thing, it has to be, no doubt there are drug co-crystals also, but generally co-formers are, so co-formers are solid liquid at room temperature, your product will be solvate. So it is just a definition part. But main thing, it should favorably change the property which you require. For example, in acid, because structure and it will enhance compressibility. If you want to enhance dissolution, will it be a polar molecule? Will it increase the that will be chemical chemistry point of view, it should form non-covalent ability. So hydrogen bond forming are preferred.
Right, sir. Sir, via various platforms, we are receiving two questions which are popping up a lot of time, which says, what is the mechanism by which co-crystals increases the compressibility, number one, and what is the mechanism by which co-crystal increases the solubility?
Okay, so mainly co-crystal increases compressibility because slip lanes are induced. Again, you can go to the structural, this paper is very good. There are many papers currently from Professor Bansal's side. Okay, Arvind Bansal has published a couple of papers. There are papers from Kelvinson. Kelvinson has prepared, from Minnesota, has published on compressional properties of co-crystal. But first paper was from Bill Jones. No doubt that he has not provided Haeckel plot or other, but main concept wise, it is very good paper. In terms of dissolution, basically the energy in the same box, when two materials are there, one has they can easily. So if we go to the solution theory, the bonding between the molecules requires your energy required to break the crystal depends on that. So energy required is less. Amorphous, it is unstable. Co-crystals are stable, but compared to amorphous co-crystals, manufacturing requires little challenge. There are some challenges, not necessary. And there are regulatory, main thing is regulatory aspect, which is the most important, and which you will be discussing in next Professor Dr. Mishra's lecture about regulatory aspect.
All right, sir, one more interesting question we got. It's again about the co-crystal. Do you face any challenges while determining the pharmacokinetics of co-crystal? Are they troublesome?
Yeah, so in terms of pharmacokinetics, so this co-crystal, what we discussed today is an excipient co-crystal. Again, this is first excipient, means not first. I'm using not went away, tried to escape from drug co-crystal because of regulatory aspect. So, but we published, I did SNDT when Dr. Kamalinder Singh was in India. We did some, no, it was not co-crystal, sorry. With Professor Nangia, we did one co-crystal on CAPE, capic acid phenylethyl ester co-crystal, and we have shown how pharmacokinetics or bioavailability increases. Pharmacokinetics is different. There is no issue because at the end you are doing HPLC separation. And there are many papers recently. When we published, it was early. Professor Nangia said, "Oh, let us do this," and we did that animal work that time. But there are published now. Pharmacokinetics is not a major challenge.
Thank you for giving such a wonderful talk and especially answering to the queries of the participants, which was very informative.
Thank you.
Yeah, I request all of you to please join me for giving huge round of applause for Professor Anand once again.
Thank you very much. Thank you. And I really enjoyed the talk today and the questions. And please be in touch, be in contact. If uh, I'm open for any collaboration, any of you want to visit Bradford, uh, if there is any funding application, you need any support from me on any funding applications, studentships, anything, please contact me via email. My email is shared on this. I'm going to share this with, I will send it to Rakesh now. And at the same time, I'm available. I use Facebook, WhatsApp, all social media. So follow me anywhere and contact me, LinkedIn. Yeah.
Thank you very much, Professor.
Thank
Very
you.
courteous of you. So kind of you, sir. So
Thank you.
now I think I invite Dr. Devraj Ram, my colleague, to introduce our second speaker, Mr. Arun Mishra. Mr. Devraj Ram, please.
Very good evening, and once again, a very warm welcome to everyone. It's my pleasure to introduce our second speaker for today's webinar, Mr. Arun Mishra. He is currently Head Global Nutrition Regulatory Affairs India and Head Global Health and Wellness Regulatory Affairs. Mr. Arun is a senior regulatory expert. He brings a strong, diverse international regulatory experience of more than 20 years of India, Europe, and emerging markets, including Asia Pacific. Previously, he worked as Executive Vice President, Regulatory Affairs for GSK Consumer Healthcare. Prior to GSK Consumer Healthcare, he was heading regulatory function for Abbott and Senior Director Regulatory Affairs Emerging Markets for GSK based at headquarters in UK, where he was responsible for developing and leading regulatory strategies for emerging markets and Asia Pacific. Mr. Arun has also worked for Pfizer Global R&D in UK and as a head regulatory affairs for Pfizer India. Arun has successfully led global regulatory strategies for a number of NCEs that are blockbuster now. Mr. Arun is on various national committees to shape the external regulatory environment and visiting faculty for various national and international institutes. For example, he is a member, scientific body, Indian Pharmacopoeial Commission, Ministry of Health and Family Welfare, Government of India, co-chair, Health and Wellness Committee, Associate Chair, OTC Committee, FICC, CCI, visiting faculty, IIMA, visiting faculty, NIPER-Ahmedabad, and visiting faculty, Center of Regulatory Excellence, Duke NUS Medical School, Singapore. With this brief introduction, it's our pleasure, sir, to invite you for this webinar. Sir, over to you.
Hi, good afternoon. Thank you for your kind introduction. Are you able to hear me? Okay. And you can see me well?
Yes, sir, very well.
Very good. So let me share my, first of all, slides and screen, if technology works. And do confirm when you are able to see my screen.
Yes, sir. Now it's visible, sir.
Are you able to see my screen properly?
Yes, sir. Sir, can you zoom in?
Better?
Yes, sir.
Very good. So thank you very much, first of all, inviting me. Thanks to NIPER-Ahmedabad. NIPER-Ahmedabad is very close to me. And I have been working with Dr. Kalia quite closely on different parts of curriculum and the program as well, and Rakesh ji as well. So thank you again for inviting. It's a pleasure to be here. And it was pleasure listening Professor Paradkar as well, complete deep in science. And I was very happy to see the patent and the collaboration that he talked about. Unfortunately, not to disappoint, I will not be talking about regulatory today. Of course, that's my expertise. But what I'm going to talk about, that why do we need to work together as an industry, academia, regulator? Why do we need to work together? especially in pharmaceutical space. And what I'll take you through in next 35, 40 minutes, I guess that's what I have, is trends that I see. And those trends are the opportunities that how do we work together and bring value for the society. Like, again, Dr. Paratkar explained an example where industry and academia came together and brought value from healthcare and wellness point of view. So again, with that, question is, why do we need to work together? Because I think together we are better than working alone, and that's the trend we see. And I'll go through various trends, and you'll see how lines are blurring, and therefore we need to come closer in India as well. If you look at overall objective from medicine, treatment, or healthcare solution development point of view, our objective is more or less the same: safety and efficacy of the medicine. That's one prime objective. The second is faster access to medicine. So the new therapies, new tools, new medicines are faster to patients or consumers. Optimizing return on investment as well, because once you have return, then you can invest further in the new therapies, new research areas. And last but not least is optimization of medicine supply, which means right quantity of medicine at the right time at the right place. There's no point having stock out situation at one place and overstocking and expiry of stock at one place, because then it's just waste of resources. And the key focus area, if you look at from any drug development point of view, remains the same: appropriate doses, route of administration, and instruction of use. Regulatory safety and efficacy requirements for key markets, including pediatric development that you are looking at. Any development will also focus on data to support on optimal access and healthcare reimbursement. In many markets, drug products are reimbursed or health solutions are reimbursed, and it's important that you factor in early enough. And commercial manufacturing strategy, because once you develop the product, then it goes globally, and you need to really understand that how you are going to supply that product to the world. And then also you need to consider life cycle requirements, additional indications, doses form, country specific requirement, and so on. So objective is very clear in terms of healthcare solution development point of view, and the focus areas are clear. What we'll talk about is some trends that we see in industry and market. This is a very typical sort of generic model of R&D and commercialization in pharma industry. You start with negative because you really have to have heavy investment when you are embarking on drug development. And then there is a period of investment, and you have product launch, you have product developed, phase 3 studies, and so on. And then slowly product uptakes happen. And slowly products reaches to break-even point. Very few products, in fact, which are successfully registered, they are able to return money. And therefore, not necessarily all the products developed throughout R&D is going to be successful and is going to return the investment. So it's important to keep in mind. And then what happens that slowly it will peak the sale, and then from business point of view, sale point of view, it will start going down. So it's a typical sort of curve of generic model of R&D and commercialization of any product that happens. The trend we see, if you look at each and every example, and I wouldn't like you to go through every line, but what is very clear is that companies are moving from massive to lean. I remember that pharma companies, big pharma companies, biotech companies, they used to have everything under one roof, from discovery to development to manufacturing to commercialization, everything under one roof. However, what we see is that many of these companies are moving towards becoming very lean company. and having focus on either therapeutic area or technology, or you see some vaccine company only. For example, GSK segregated the consumer healthcare business and has announced to spin off that business eventually and make it a standalone business. So you see many companies, they are going in the same direction from massive to lean. That's one trend to keep in mind, and we'll come back to that, why it is relevant for us. The other one is very clear. What we see as a trend is that companies are moving from hub to hotspots. As I said in the past, big pharma companies and even mid-sized pharma companies, they had R&D footprint in terms of hubs. So they had, for example, Pfizer had in US, they had in China, they had in Europe, quite big hubs. And slowly, what we see as a trend is converting into hotspots. So not necessarily then you are having everything under one roof as a mega organization, but then you are looking at smaller hotspots in different countries and linkage within those hotspots as well. So that's another trend that we clearly see from R&D point of view in bigger companies. Interestingly, as Professor Paradkar was talking about as well, is that we believe that only big pharma companies could invent and discover and develop new medicines, but that's changing. What we see more and more is industry, academia coming together, and they are developing new medicines and new therapies. For example, very recent one where AstraZeneca, Cambridge, and Serum Institute came together and looking for corona vaccine. And it's a perfect partnership of manufacturing ability, R&D ability, capability, and science and technology at the same time. So it's a perfect example again. Similarly, what you see on the screen is, again, a great example of university, academia coming together with a small company and really bringing therapies and new drug development as well. So again, that's the trend we see. What we also see more and more is co-location of industry and academia. So for example, in the UK, you have Cambridge Research Park, where industry have established the R&D center. Similarly, you see in US, Boston is a hotspot again, where industry and academia comes together. So that's again another trend, co-location of industry and academia to really harness the capabilities and abilities of these two institutions to advance the science and therapies. And again, something perhaps NIPER-Ahmedabad is well suited to be in, and Gujarat has been pioneer when it comes to pharmaceutical manufacturing technology development. And therefore, I don't see any reason why NIPER cannot be the pioneer is in similar model where bring industry academia together and co-locate and development new medicines and drugs. The other trend we see is primary light but specialty heavy. Majority of the examples that you see on the screen and in the market as well, you will see the companies are moving to a biologics, biosimilars, and I'll talk about gene therapy and cell therapy as well. But that's another trend we see. And what you see is, again, is sort of patent expiry time frame, and you will see a lot more biosimilars of these molecules that are coming. And again, it's a very competitive area, very profitable area as well for business. And again, bringing therapies which are relevant from patient point of view. Let me move on to the next one. I think the other important trend I see is moving from west to east. In the past, what we saw that drugs were developed for US and European markets. And then most likely it was taken to other markets like India, China, and emerging markets and so on. But today what we see, because business is really gaining momentum in emerging markets, therefore these markets like India, China, and many others, they are coming in the center of development and consideration for big companies as well. And it's very clear if you look at the growth rate. For example, you'll find China, Brazil leading in terms of growth rates, even beyond US. Of course, by size of the market, US is still far biggest market. But if you look at growth point of view, you're looking at China, India, Brazil, Russia sort of market from pharmaceutical business point of view. So again, the trend we see is west to east and much more comprehensive sort of approach in development. And therefore, again, institutions like NIPER and many other in India becomes much more relevant in partnership to develop and come together with the companies to develop therapies which are much more relevant locally for us as well. So those were in terms of R&D sort of trends that I see, and why it is relevant and important for us to come and work together. I would like to talk about some trends around the delivery system as well, healthcare delivery, and how that is shaping and changing, and why, again, it's a food for thought for all of us, is that why it is important that we work together again, and what are those areas we could think of again to come together. And at the end of this discussion, I would share my thinking that perhaps a framework how we could look at it for India. I thought this was very appropriate picture as well. What we see more and more is that power is shifting. Power is shifting to patients and to consumers, from physicians to pharma companies and medical device companies. And it is very critical. If you look at simple examples, in the past, if you have, let's say, fever, you go to doctor will examine, will give you medicine, you take the medicine, and if it's not done, you go back to doctor again. By and large, still happens, but what we see growing trend is that if you have symptoms, you do a Google search first and find out what's wrong with you and try to understand what has happened. And then try to understand what potentially could be the solution. And then you have platforms like Practo and many others. Then you look for a consultant or doctor who you could go to. And in nowadays, in COVID days, you do electronically do a video call. You don't have to even go. And then you ask 1,000 more questions around the disease, around the treatment, and so on. So that's what is shifting. The power is shifting to patients and consumers, to payers as well, and to data platforms. And that's very relevant and important to understand. Now, as I said before, the lines are blurring. In the past, you would have gone a prescription, got a data, gone to a lab, pathological lab, got your blood drawn and data analyzed, and then gone to doctor back, and doctor would have looked at it, what is wrong with you in terms of maybe your vitamin D was low and your B12 was low, or hemoglobin was low, or uric acid was high, and so on, would have done the assessment and recommended you something. But look at what is happening today. It's a real-time data. In fact, Apple Watch now has ability to constantly monitor your ECG and really give you alert in terms of if you have any cardiovascular event risk at the same time. And that's just an example. Today in Corona days, you would have looked at everyone has oximeter and knows what oximeter is, how to use it, and what does it mean. Similarly, blood pressure, more or less every house has blood pressure machine and thermometer if you have elderly at home. So point is that consumerization of healthcare, consumerization of data, accessibility of data, and reducing the lag and measure is really empowering real-time decision, not only for the consumers, but healthcare practitioner as well. And therefore, lines are blurring. Where do you draw the line between diagnosis, treatment, sort of pendulum, if you look at it? And therefore, do we see an opportunity? And I see, again, India fits very well, being the IT power of the world, medical power of the world, pharmaceutical power of the world. I think there is an opportunity for us to really lead the world when it comes to integrating technology, science, and data, and really making public health care much more accessible, affordable, and meaningful from public health point of view. I think something to think about for us and institutions like NIPER, that is there something that we could lead in this direction. I thought this was very interesting example, again, the trend. So Larry, what he did, he is a founding director of California Institute of Telecommunication and Information Technology. What he did that he created 3D, digital 3D version of himself. And then when he went for surgery, he invited his doctor and said, and took her in his VR cave and explained her where is the problem and where surgery should be done. And then resultant surgery was much shorter and it was much more precise, and it was robotic surgery. So look at again how the science, data, technology is blurring and coming together, and therefore bringing value to patient and consumer and health, public health outcome again. Again, I think institutions like NIPER, IITs, AIMS are very well placed to come together. And question for us would be, how do we create the ecosystem where we draw the, really break the boundaries and really blur the lines of different disciplines and bring them together to really merge these powerful technologies, sciences together for public health point of view. Something to again think about. And I fundamentally believe that NIPER-Ahmedabad and institutes like IIT and AIMS could take a lead and bring value again, not only for India, but for around the world. Again, fantastic example. Recently, FDA approved Digiceutical. This is the first Digiceutical approved by US FDA, and it is a prescription therapy. It's a cognitive behavior therapy. Again, there is no pill involved. It's like a video, sort of 3D video game. You put it on your eyes, you have time period to watch and frequency to watch, and that's it. So think of again that how lines are blurring when it comes to technology and therapy in this case as well. And again, it's a path breaking. From regulatory point of view, it's a challenging as well. It's a prescription therapy. How do you then therefore look at efficacy, safety, quality in traditional way? And therefore we need to switch gears and find a different way to evaluate these products and really allow to the market and bring it to healthcare. And that's not only one. If you look at number of efficacy studies being done on digital therapy is huge. And therefore you will see more and more digiceuticals coming in the future. And as a country, regulatory framework, clinical framework, therapy framework, we need to understand how we are going to adopt this and really bring value to the patient and consumers as well. But not only that, I want to step back again to look at again, putting this to NIPER as a challenge, that how do we take on these early on these waves and bring value not only for the country, but for the world. The other trend we see, which we never saw before, is pay for the results. And that's where you are moving into precision therapy, sort of gene therapy sort of space, where of course cost is, for example, the Novartis treatment cost is around $500,000, but it's a money back guarantee that if it doesn't work, and it's a cure, and if it doesn't work, your money is returned. And that's where it's going. So society and the healthcare framework, the payers, the decision makers will ask more and more precision, more and more results, more and more guarantee that it's going to work. And therefore, we need to think that from technology, science, R&D point of view, how do we need to change, and how do we need to work together? In this case, for example, not only necessarily academia and regulators, but also You are looking at payers and healthcare decision makers to work together to bring therapies which are much more precise and guaranteed in nature. And I talked about briefly on gene therapy. Again, it's not a fantasy anymore. Gene therapy is reality, and we have seen that, and it's a promising. Of course, there are a lot of challenges. There are a lot of innovation area. There are a lot of opportunity to improve from R&D, manufacturing, supply chain point of view, but it's reality. And you would have seen multiple examples. If you Google, you will find it. I think the question is, can we turn this into medical products and can we manufacture it? Because so far, what we have seen is a lab base. But we have advanced, we have now progressed. And yes, we have treatment now towards rare diseases, ultra rare diseases, sort of large unmet need area. Question is, on the path that we are traveling today, can we expand into in vivo therapies and new cell types? And therefore, can we take it to the mass level? I think that's the direction we are going to follow. I think important area would be efficacy, cost of goods. How do we bring it down? Therefore, there's a greater access and there's a greater feasibility for other diseases and wider diseases as well. So I think that's another emerging area that we need to think, can we be on the forefront of science and technology when it comes to India, Indian institution and power of science that India has? Let me see if it is, oh, sorry, it's going back. Right. So we talked about, as I said, yes, it is possible to make it commercial reality, gene therapy. And we have seen whether it's a Novartis example, Gilead example, but it has been on in the news. But if you look at more from science and technology point of view, there are many challenges. Adaptive cell gene therapies are highly expensive at the moment. Question is, how do we bring it down? Traditional pharma pricing model may not be appropriate. We need to find alternate pricing model, alternate reimbursement model. It's a challenging for reimburser as well because it's a new sponsor, it's a new payer. However, it's clear that there is significant need of innovation in manufacturing, especially around vector cell processing and logistics. And there is innovation required in safety improvements of these products. But again, it's very promising. Gene therapy is promising and it's really helping patients. Real world evidence, I'm conscious of time as well. So I'll briefly talk about, I will not go into detail. During the drug development, we look at a lot of data coming from our clinical trials, whether it's a phase 2, phase 3 trials. And then once the product is approved, then it goes to the masses. And then in terms of pharmacovigilance, we start collecting data. I think the question is that now we have better accessibility and connectivity of the data. How do we make a sense of it in terms of having a better evaluation of any therapy from safety and efficacy point of view? And that's the question we have. And that's where real world data and therefore real world evidence comes into the picture. Real world evidence is simply that what do you make of real world data? How do you analyze and what information you get? And how can you use that information to convert that into knowledge and therefore decision-making process? It has been in the news for long and rightly so. But I think if you look at in pharmaceutical industry, real world data and real world evidence is already used for decades, but more towards the right side of the spectrum, not necessarily towards left. When you look at new drug approval or labeling revision and expansion of products, that is mostly based on phase 3 studies, phase 2 studies, or let's say randomized clinical studies or controlled clinical studies. However, if you look at the right side of the spectrum in terms of post-marketing surveillance point of view, that's where you would see routinely real-world data and real-world evidences are used to make a decision. You would have heard many a times that products are recalled, and that is based on real-world data and evidence that you're looking at. But there is increasing demand to move that spectrum towards left side, towards new drug development, new indication, and so on. I will not go through bullet and bullet, but there is increasing demand and there are reasons, and there is increasing supply as well because of the connectivity that we saw. I think the question is that how do we manage and what it is used for different players. So regulators will have more comprehensive risk benefit assessment. They will have much more flexibility around sequencing of evidence. For payers' point of view, they will have much more comprehensive value assessment of medicine's lifespan. Care delivery and resource utilization planning would be better if you have real world evidence data. It will support outcome-based contracting if you are contracting out. It will assess patients' adherence and discontinuation of treatment if needed. And it will also assess clinical adherence of treatment guidelines. And for clinician and patient point of view, it will provide insight on how well the treatment might work for an individual patient at an individual level. And it will regularly update care pathway based on the data from real world evidence is coming. So therefore, there is a value for each and every stakeholder in the spectrum when it comes to real world data and evidence. But there is a hesitancy. And there are question marks around data quality, real world evidence science, and experience and understanding. Quality sources are fragmented. You get it from website, you get it from Facebook, let's say, you get it from Google, and so on. Often data is incomplete. You don't have enough data point and full data to really assess and homogenize the data for any assessment. Non-standard taxonomies for the same thing. Someone is saying diarrhea, someone is saying something else. So it's a non-standard taxonomy used in real world data. And there is uncertainty in reliability of accuracy of the data at the same time as well. From design and method point of view, the science is still evolving that how do you really remove any biasness without any randomization? Is there a standard process that we can evolve? And lack of transparency of data sources as well. And therefore, there is a limited experience working with real world evidence and lack of expertise as well. Why I wanted to bring it here is that, again, if you look at India is known for mathematical modeling, known for statistical design. It has been a hub of clinical development. It has been a hub of pharmaceutical development. Institutions like, again, IITs and NIPERs could really take a lead in real world data and real world evidence and bring that value to pharmaceutical industry and to not only for India, but to the world, and really pioneer this space of real world data and evidence and bring those and focus on those areas. For example, how do we reduce the biasness without randomization? Because in randomized clinical trial, we can easily do that. How do we standardize the process to assess the data and make any sense of it? Similarly, lack of transparency, how do we improve transparency? So again, it's another stream which I believe that institutions like NIPER is well positioned to take the lead and really bring industry academia together. I'll skip that part and I'll briefly talk about COVID-19 trends that I see. It is very clear that industry, academia, and regulators are coming together to really fight against the crisis. Around the world, we have seen the regulators and policymakers, how swiftly they have been updating and changing, amending the policies to bring the tools that are required to fight against the crisis. We have seen how academia has really quickly looked at scientific part of multiple solutions, whether it was around sanitizers, whether it was around repurposing of medicines that we at CQ we looked at. And at the same time, we also have seen how industry has really come together, whether it's the vaccine development or repurposing of medicine to really bring those solutions. Again, fantastic example and demonstration that how industry, regulator, and academia has come together in crisis to really join hands together and continue our fight against the crisis. And I'm hoping, and this is a sign we should take learning from that, that we should continue this collaboration in the future as well to bring those solutions which are required from public health point of view. Some trends that I saw, as I said, regulatory industry partnership, it has becoming a part of solution. We have seen emergency approval processes, accelerated review processes. We have seen much more enforcement as well. We have seen many claims made around the world by opportunistic brands and companies, but then regulators have stepped up and enforced. And we are seeing more and more regulator partnership among regulators as well. Even I see in India at the state level, for example, Kaushaji in Ahmedabad, Gandhinagar has been pioneering many areas, for example, hand sanitizer, how Gujarat became the supplier of hand sanitizer in the country, where we were struggling even to supply in Ahmedabad only. So again, we see a lot of partnership and opportunity there that we need to continue. I think in summary, that's my last slide, is that there is the current pharma industry R&D point of view is going through significant change. Patient consumers are taking more control, and there is increasing transparency, partnership, accountability on healthcare and its delivery. Lines are blurring between the technologies, between the stakeholders, between the pharma, academia, company, and regulator players, and even the big data and tech companies as well. Responding to that is global regulatory framework, which is evolving and responding to the science and technology as advancement and supporting innovation, development, manufacturing, and commercialization of new healthcare solutions. And companies are developing optimized products specific to global regulatory strategies to maximize patient benefit. And academy, industry, and regulators need to work together to advance healthcare agenda. And I firmly believe, I will restate again, is that NIPER-Ahmedabad is in the right place to lead this for India and bring industry, academia, collaboration, partnership as a model, role model for the country and for the region as well. With that, I really thank again for NIPER for inviting me, and also I'm thanking to the director of NIPER-Ahmedabad for having me today. And let's stay connected. My email address is on the screen. If you need anything, I'll be more than happy to help you out. With that, thank you again, Rakeshji. I'm happy to have any question if there is any.
Thank you, sir. Thank you so much. It was really wonderful talk, and we feel blessed after listening you. Sir, with your permissions, there are some questions which are raised by the participants. With your permission, can I narrate those questions? Hello, sir. Hello, sir.
Yes, please. Sorry, I was on mute.
Okay. Sir, thank you for the wonderful talk, and we are really blessed after listening you. Sir, with your permission, there are some questions from the participants, and with your permission, I would like to narrate those questions. Sir, can I go ahead?
Please.
Yes, sir. Sir, the one question is that why, almost in all the instances, a scenario where, we see the first time the drug is approved in the US and subsequently it is approved in the other markets like Europe and the other emerging markets. So this is the general scenario. What could be the reason?
So I think there are multiple reasons. One is that countries like United States and European countries, they have heavily invested in R&D and look for newer therapies. And therefore, you have R&D centers there, you have capabilities there, and investment at the same time as well. And therefore, you see the therapies being developed in those countries first. Plus, regulatory authority and regulatory framework is equipped to assess new drugs and new therapies. However, said that, it's changing now. We are seeing more and more emerging markets, countries like India, China, and many others. They are advancing their R&D efforts. And for example, even in India, you look at many companies like Glenmark and many other I have seen, they have very healthy pipeline of NCE development. Of course, it's substantial investment and risk as well. And therefore, you see multiple partnerships happening in India as well to advance those drug development. So I'm very confident that countries like India, China, Brazil, and Russia, for that matter, will advance those therapies and will bring NCEs or new drugs, which are much more relevant, not only for India, but for global health as well.
Can be done to bridge the gap between academia and industrial research?
I think that's a very important question, and it's $1 million question as well, that how to do that. And I think we just need to take baby steps in many ways. I think one, I believe, is that the recent revision of science, technology, and innovation policy, which is driven by the framework, is really helping to bring industry and academia closer. That's one. I think second is that the curriculum of institutions like NIPER and many other perhaps could be much more an opportunity to really understand industry better from early ahead in their student journey or educational journey, and also get opportunities of internships so that they understand the mindset, the framework, and the approach. I think that's the third thing I would think of. And I think the fourth one, which is, I think, very important and critical and could work, is a co-location. It's a model like a Cambridge model, Boston model, where we could bring industry and academia co-located together and really work hand in hand and blur the line between two of them and really make it one effort. And again, the objective of these both is to have better public healthcare solutions. So I think these, in my view, are 3-4 areas that perhaps bring academy industry closure, because it's not that in India we don't have good scientific output. I think where we face challenge is that those good scientific outcomes are not translating into public health solutions. I think that's where we need to find a way that how do we translate those science into public health solution, i.e. into products, into drugs, into devices, diagnostics, and so on. I think that would be the fourth. The fifth one, which I almost forgot, is I think NIPER is doing, but we could do more, is to start creating incubators for startups and ecosystems for startups, where industry, academia, government could come together and really help startups and incubate startups so that they could venture in that direction and bring something completely new and fresh minds as well. So I think those would be 5 sort of spaces I would think of where we could bring industry and academia together, and importantly, regulators as well.
The next question is that, what are your views on gene therapy for lifestyle disorder like obesity and cardiovascular disorders?
Look, I would go back to what I said. Gene therapy is something very new and very promising. And the areas where it could help from public health point of view is endless. I think it's more of scientific advancement and how do we bring solution and looking into those areas whether it could work. So in summary, I think in principle, yes, but we really need to travel that path to reach to the solution level. I think you are on mute.
Okay, so now, am I audible?
Yes.
Okay, so the last question is, what are regulatory aspects for digital and personalized therapeutics?
That's a very important and interesting question. Tell me how long I have, because I could talk hours and hours on that. So whoever has asked the question, I don't want to waste everyone's time. Of course, in summary, personalized medicine and digital health or digital therapy is an evolving area from science and technology point of view. Therefore, from regulatory framework point of view is also evolving. For example, India doesn't have digiceutical regulation yet. Personalized medicine again is evolving around the world. But if that is something area of interest, I'm sure we could plan a guest lecture or something in the future, and I'll be happy to talk.
Thank you, sir. Thank you so much. And sir, we wish that we want to hear you once again in the future also. Thank you so much, sir, once again
Absolutely.
for giving us time and this wonderful talk.
Thank you very much. Thank you for having me. Pleasure was mine. I hope it was useful for everyone, and I wish all the best. Thank you.
Thank you so much.
Thank you. Bye-bye.
Bye-bye, sir. Moving to the third speaker, it's my pleasure to introduce Professor Nizar Al-Sari. Professor Nizar is a faculty at Department of Medicinal Chemistry and Drug Design, Jordan University of Science and Technology, Irbid, Jordan. He earned his PhD in Computer-Aided Drug Design, University of Strathclyde, Glasgow, Scotland, UK. He did M.Pharm in Medicinal Chemistry from Just, again Jordan. Currently, he is serving as a faculty, Jordan University of Science and Technology, Jordan. He served on several pivotal administrative and executive posts, including Vice Dean for Accreditation and Quality Assurance. He played a crucial role in many councils and high committees. Professor Nizar is also a reviewer for many peer-reviewed journals of international repute. He has more than 50 research papers and conference presentations to his credit. He also honored with many prestigious awards. With this, now I invite Professor Nizar to deliver his talk. Sir, over to you.
Okay, can you hear me clearly?
Yes, sir.
Okay, so first of all, I would like to thank the organizing committee for preparing this event. And in particular, I would like to thank Dr. Rakesh for his kind invitation. And definitely, we need to thank our previous eminent speakers, Professor Anant and Dr. Aaron, for their insightful, informative talks. Since the theme of this event or this webinar is the interdisciplinary collaboration in scientific research, I will be addressing this topic by shedding some light and discussing some of my work pertinent to COVID-19 pandemic, which unfortunately has affected the lives of all people around the world. And the motive for this work actually was based on some kind of moral issues. I'm a medicinal chemist, I'm a drug designer, so I thought it was a obligation, some kind of obligation to participate or contribute even a little bit to the global efforts in fighting against this serious disease. So before we move on, I would like to share a few points that I would think will frame the essence of interdisciplinary collaboration. So this is actually the pipeline of drug discovery in terms of pharmaceutical research. We can see that there are different phases that any successful drug need to pass it through to reach the market, starting with drug discovery to preclinical trials, the clinical trials, and regulatory approvals, then to the market. As we can see, around 5,000 to 10,000 compounds are being tested, out of which only around 250 compounds can enter the preclinical trials. And the successful ones that can reach clinical trials on humans are not more than a handful, maybe 5 or less. And ultimately, one of them could be successful and reach the market. This is the general idea. So by looking at the different phases, we can realize that no one can do the whole process by himself. So the age of one-man show, I believe, has far gone away. So now you need to collaborate with other people, with other expertise from different disciplines in order to, in order to make the success you are aspiring to. The other two important facts that will open the avenue for my talk are the next two things, which are the time and the cost for this whole process. As you can see, the whole process from drug discovery and to the development up to the point where the drug, a successful drug, can reach a market can take roughly from 10 to 15 years of dedicated hard work. And the capital cost for this process can reach up to 2.5 billion US dollars. So if there is any way that can you cut down the time or the expenses for this lengthy, expensive process, then you will have a breakthrough. And here comes a new advent, let us say, in the field of drug discovery and drug design, which is the application of computer-aided drug design. Since the introduction of this technique, the enrichment in this process has, in some cases, reached up to 20%. So if we would like to translate this into time and money, we can save up to 2 years, and you can save up to 500 million US dollars. And the good thing about that, for any drug discovery project, at some point you need to protect your idea. So you file a patency, and roughly the time that will be allocated for this patency is around 15 years. The good news is, once you have a successful drug that can reach the market, all these expenses can be returned in roughly around a year. So if you manage to extend the years of your protected patency, then your revenue will be much higher. So these are the basic things that I would like to share before going to my talk. And given the nature of the audience, since it is spanning a different spectrum from students to PhD students to postdocs, I try to my talk to make it starting from a very shallow curve and steeping up along the way. So I would like to give a very brief introduction about the methodologies that I've implemented in this work. So in this introduction, I will give a very brief idea about the COVID-19, which unfortunately now every one of us has a lot of information about this pandemic disease. And I will shed some light on the structure-based drug design technology, which is now streaming the drug discovery processes, and MD simulations, which is also a very handy tool in terms of studying biomolecular compounds and biomolecular systems. So let's move on. COVID-19. The basic idea that I would like to highlight about COVID-19 is the process of invasion, cellular invasion, and what happens when it's inside the cell. So here we have the COVID, the SARS-CoV-2, or the COVID-19 virus. It is an RNA virus, single-stranded RNA virus. And once it reaches the host cell, let us say a lung cell in a human body, it will attach itself to the receptor, which is the ACE2 receptor. And by this, it will gain access to the cytoplasm of the cell. Once there, the envelope will open to let the single-stranded DNA get integrated with the components of the human cell. And afterwards, it will be translated into long polyproteins. Those long polyproteins are non-functional. So in order to be functional, so that the virus can reproduce itself and invade other cells, they need to be processed. The major enzyme that is responsible for processing these polyproteins and generating functional peptides is the COVID-19 maniprotease, which is the focus of the current talk. So by the action of this enzyme, these long polyproteins will be cut down into smaller functional peptides, and these functional peptides will help the virus to assemble a new viral particles that can easily invade budding of the cell and invade nearby cells. So theoretically or basically, if we manage to stop or block the function of the COVID-19 maniprotease, then we will break the cycle and we will treat any infected person. So the good thing about this enzyme, it is a unique enzyme. It is only available in the virus. So us as humans, we don't have this protein. So if anyone managed to design a selective and specific drug that can inhibit the function of this protein, theoretically, the side effects of this drug will be minimal to humans. So hopefully, there's now a lot of research going all over the world, targeting these specific proteins. I'm just talking about one of the major proteins. There are other proteins specific to the COVID-19 virus that are under intensive research, aiming to identify new therapeutics for this pandemic disease. And one of the first approaches that have been undertook was repurposing already FDA approved drugs because they have known, let us say, pharmacokinetic and pharmacodynamic profile, and the toxicity profile is very well known as well. So this was the first idea that most researchers around the world has tried to combat the COVID-19. So this is the general idea about the invasion. Now let us have a deeper look about the structure of the target itself, the COVID-19 main protease. As you can see here, the crystal structure of the protein, here is a cartoon representation of the protein. We can see there is 3 domains. We have domain 1, domain 2, and domain 3. Domain 2 and 3 are connected by a long loop. And here is the active site of the protein. Here is the cutting of the polyprotein happens. So this is a peptide segment bound to the active site of the protein. This is a surface representation. And here we can see a close-up view of the active site and the different sub-buckets that constitutes the active site. So basically, the catalytic amino acids that are available in the protein, we have a cysteine and a histidine amino acids. This catalytic dyad is important for the proper function of the protein. So this is, in general, the 3D structure of the protein. As I move on during the coming 30 minutes or so, we will have a deeper look at the binding site and what is happening there. How can we utilize the information that we can gather from this structural data to tackle this pandemic disease? The other thing that I would like to introduce in a very, very abstracted, simple way is the structure-based drug design concept and the computer-aided drug design. Since the advent of, let us say, supercomputers, high-performing computers, and the efficient algorithms, now computer-aided drug design became an integral part in any drug discovery process. And basically, we can divide the approaches that can be undertaken under this bigger umbrella into 2 main approaches. The first one is structure-based drug design. And this is the case when we have 3D crystal structures for the protein of interest. In our case, for example, the COVID-19 main protease, we have dissolved the crystal structure. These crystal structures could be obtained from X-ray crystallography, NMR data, or from homology models. So once we have the structure, we can design compound or search for compounds that can specifically bind to the active site of the protein and block it. The other approach is called the ligand-based drug design. In other cases, we might have don't have the 3D structure of the protein, but we have a number of known compounds or known inhibitors that are targeting that particular protein. Based on these ligands, we can generate a negative image of the binding site. So we have different applications that can follow each approach. For example, in structure-based drug design, we can use the de novo drug design. De novo means from scratch. And since the ligand-target interaction is based on complementarity, and this complementarity is two terms, let us say, the shape or the geometric complementarity and the electronic complementarity. So if the ligand would like to bind to the active site, then there should be good complementarity between the two. So if you have the shape of the binding site, you have a good definition of the binding site, you know exactly which amino acid is catalytic, which amino acid is a binder, you can design a compound that specifically interact with each of them to end up with a highly selective drug. And this is the de novo design approach. Another method is the pharmacophore modeling. Pharmacophore modeling, in the next slide, I will elaborate a little bit more about it. So in pharmacophore modeling, you generate 3D pharmacophore as a search query that can be used to virtual screen large databases of drug-like compounds. And the third main approach is the molecular docking. And I also will elaborate a little bit more about that because it is one of the essential methodologies implemented in this research. For the ligand-based design, also you can generate pharmacophore models based on the knowledge that can be extracted from the actives and inactives. I mean the active drugs that inhibit the particular enzyme that we don't have a 3D crystal structure for, and the inactives that can represent bad complementarity, let us say, in terms of shape or electronic. Also, we can study a quantitative structure-activity relationship. And also you can do some kind of similarity search, because one of these compounds, for example, is a very potent inhibitor. You can search the chemical space for relatives of this compound that can show even more better pharmacokinetic or pharmacodynamic profiles. So I will focus a little bit more on pharmacophore and molecular docking. Then I will move into the specific issues of the work itself. So pharmacophore modeling. What do we mean by a pharmacophore? Simply, it is an abstracted description of the molecular features that are necessary for molecular recognition between the protein and the ligand. We just said that the binding is determined or defined by the complementarity. And we need to define a very subtle difference between pharmacophoric feature and a chemical feature. For example, if you have a carbonyl oxygen, it is a carbonyl in terms of chemistry, but it is a hydrogen bond acceptor in terms of pharmacophore modeling. So this pharmacophore actually represents, because we are talking about the complementarity, it is more like a negative image of the binding site. And because this, if you would like to use it as a search query in virtual screening, the efficiency will be much higher compared to docking studies, because it will act like a mold, and then you can fit all the ligands in the database. And if a ligand can fit the pharmacophore, you can save it. Otherwise, it will be filtered out. So in our case, let us have a look at the different types of pharmacophoric features. We have, for example, this one is an aromatic ring or ring aromatic feature, a hydrophobic feature, a positive ionizable feature. For example, if we have an amino group, here we have a hydrogen bond donor. For example, in this case, we have an amide, or a negative ionizable like a carboxylate or similar functionalities. Also, we have hydrogen bond acceptors. So if we would like to use this as a tool for virtual screening, how can we differentiate between good ligands and bad ligands? We need to have a value. This value is the fit value. So if we have, for example, a pharmacophore, like the one we are seeing right now, we have 1, 2, 3, 4, 5, 6 features. So if this ligand is matching the center of each of these features, then the fit value will be 6. And if one of these features, for example, if this one is not mapped, then the fit value for that particular ligand will be 4. If 2 are missing, it will be 5. And if 2 are missing, it will be 4, and so on and so forth. This is one thing. Another thing, if my hand is the center of the pharmacophoric feature, and there is some kind of deviation from the center of these features, again, the fit value will drop. So we can use a threshold. Usually it is an arbitrary threshold for the fit value, based on which we can filter out all bad binders. So this is briefly what do we mean by pharmacophore, and how can we use it for virtual screening of commercial or small molecule databases. Now let us elaborate a little bit more about the molecular docking. And by the way, molecular docking is an integral part in any structure-based drug design study. So basically, we have the target protein and the ligand, or a set of ligands, and we need to try to fit these ligands in the active site of the protein. And based on that, for any docking algorithm, we need to have 2 components. The first one is a search algorithm that can try to find different poses of the ligand within the active site. And the other part is the scoring function that will give us an approximation of the binding affinity of that ligand. So at the end of the docking study, we will end up with different binding orientations with different scoring or representation of binding affinity, based on which we can rank these compounds or these ligands that this would be a high potential ligand and this would not be. So the docking process will be conducted by algorithms, the search algorithm and the searching algorithm. And what the algorithms will do is simply try to fit the ligands inside the binding site, trying different conformation for each ligand and score that conformation. Then the data will be tabulated and the analysis step will commence. So this is basically what we mean by the molecular docking. One major limitation of molecular docking is the flexibility of the target protein. We all know that the ligand receptor interaction actually is better represented by the induced fit, or recently a newer term is emerging, which is conformational selection and induced fit. Anyways, the ligand itself and the protein, in reality, they are dynamic rather than static compounds. So they are continuously moving. So in molecular docking, the protein is usually treated as a rigid body, and this is unrealistic. Nevertheless, it provides a lot of good information that can guide the process of drug design. But if we are interested in knowing more information, deeper insight about the dynamical behavior of the complex, then we need to move a further step, which is the molecular dynamic simulation. So what do we mean by molecular dynamic simulation? The simple definition is computer simulation of molecular system, for example, a protein or a protein ligand complex, based on the application of Newton's laws of motion, particularly the second law of motion, to describe the evolution of the system with time. So to make it much easier, I'll not go very deep into physics and math in terms of MD simulation. I will just try to explain the concept. So we would like to study the dynamical nature of the protein or the protein ligand complex, and both of them, they are moving. And the protein is made of atoms. So in molecular dynamics, it is based on molecular mechanics. And in molecular mechanics, there is a basic approximation, or this approximation, it treats the atoms as particles or balls, and the covalent bonds that connect them as springs. So MD basically is an implementation of molecular mechanics, which is completely different from quantum mechanics. So let us imagine it this way. We have a ball, and I would like to kick that ball. And I want to know, after a specified period of time, where that ball will be. In order to know the new position, I need to know the distance that has been traveled. And in order to know the distance, I need to know the velocity. And in order to know the velocity, I can obtain it from the acceleration, because all of them, they are the derivatives of each other. The acceleration itself can be obtained from the force. And here we come to the second law of motion. And if we would like to know the force acting on each particle, on each atom, we can calculate it from the gradient of the potential energy. Then you iterate as long as you want, as much as you want, as your resources can allow you. And each full cycle of these integrations is called an iteration. So basically, we need just to know the force. So how can we calculate the force acting on each atom? We need just to calculate the potential energy of the system. So is there any way to calculate it? Yes, by using force fields. But before going to force fields, this is the end product of the MD simulation. After each specified period of time, and usually it is in a femtosecond scale, we have a new 3D shape of the same protein. So this is a new conformation, a new conformation, new conformation. And by the end of the simulation, you will end up with a trajectory that describes the behavior of the system with time. So basically, to run an MD simulation, you need to have a set of coordinates, which can obtain from X-ray crystallography or NMR or homology models, then a random distribution of velocities, which can obtain from Maxwell-Boltzmann distribution. Then you calculate the acceleration or the velocity from the acceleration, and the acceleration from the force, and the force from the gradient of the potential energy, which can be calculated using a force field. So the force field, it is a functional form and a set of parameters that can be used to calculate and describe the potential energy of a system of particles. And now going back to a protein or any chemical compound, we have the atoms. So what kind of energy is affecting that atom? We have two types of energies. One is resulting from the bonded interaction between atoms that have covalent bonds, and the other between nearby atoms that are not connected, but they are near in space. So we have two components of this force field that can calculate the total potential energy based on the bonded and the non-bonded interaction. And let me elaborate a little bit further, and by that I will end up my introduction. So this is the mathematical equation of the force field. We have two components. We have the bonded terms, which are the bond stretching, angle bending, and dihedral rotation. And the other part is the non-bonded interactions, which are the van der Waals interaction. Here we have two atoms in space. Once they are approaching each other, there will be an attractive force because of the random distribution of the electronic cloud. But once they reach a closer distance, they will start to repel each other unless a covalent bond is formed. And this is accounted for or can be calculated using the linear joint potential. The other part is the electrostatic, which is very well known, resulting from transfer of electron from one atom to other, generating ions with opposite charges, and then we will have attractive forces. So the force field can calculate all of these energy values, sum up them, and based on it, once we have this potential energy, the force, the acceleration, the velocity, and the new position will be in hand. So we can simulate as much as we can, and we can calculate all the dynamical processes, properties that we are interested in studying. So YMD simulation is very important because based on the generated trajectory, you will end up with a huge amount of dynamical, structural, and energetic information. And this, in fact, enables us to understand in a much better way the dynamics structure-function relationship. Now, we all know that for any functionally protein, it has to have a specific 3D structure. And in the most abstracted way, let us imagine, for example, an enzyme. The main protease, for example. We can say that we have two states, the open active, the closed inactive. So the shape of the protein dictates the function. And this shape is governed by the native dynamics of the protein. So if you can understand the dynamics of the protein, you can fine-tune or you can block the function of the protein. And this is very important and very interesting in the same time to understand and study this molecular system at the atomic levels. And that's why in some of the literature, you might see that molecular dynamics is being referred to as being atomic microscope, because you can see what is happening at the atomic level. Now, I will give a very brief summary of the methodology so everyone can keep a track of what is happening. So the idea that I implemented in tackling COVID-19 maniprotease was based on the structural data hidden within the crystal structure itself. So the first step was exploring the protein data bank for available solved 3D crystals of the maniprotease enzyme. And there's actually plenty of them nowadays. And those crystals were actually studied they need to be complexes. And the idea behind that I wanted to identify the key interacting residues within the active site of the protein. So these are the key interacting residues. So for any of these ligands, if it is establishing good interaction with amino acids, then it will be good binder. Otherwise, during the crystallization environment, it will not stay in the active site for a long time. So these key interacting residues, because they are the very, very crucial in the binding site, and they could be even catalytic or binders, could be utilized to generate pharmacophores. And these pharmacophores, because we said that they are very excellent tools for virtual screening of large databases, and in the same time, they are representing more like a negative image of the binding site to screen large databases. Then these databases, or the retrieved hits based on this virtual screening, were filtered to filter out the bad binders or the compounds that don't comply with the drug likeness. For example, one of the filters were the Vipper rule, others was the Lipinski's rule of 5. The fit value itself was considered as a filter. So the filtered compounds were then docked into the active site of the maniprotease and rescored, and their free binding energies were calculated using the MMPPSA solvent, implicit solvent model. And the best scoring ones were further filtered by calculating their ADMET descriptors. And the ones that passed all these filtration processes and showed good ADMET characteristics with good scoring were considered as potential main protease enzyme inhibitors. And because we just said that docking itself neglects usually the flexibility of the binding site, currently the best docking algorithm can account only for limited flexibility of the side chains of the binding site. So if we would like to have a more realistic look, then we need to run MD simulation. And this is what has happened. So let us move now to discuss in a little bit more details the results of each of these single steps. So we will start with the structural analysis. As I just said, there was a plenty of available crystal structures in the protein data bank pertinent to the COVID-19 main protease. And the ones we selected 24 crystal structures. And the selection criteria was based on the resolution of the solved crystal and the quality of the crystal in terms of there's any missing loops or incomplete structures, and the nature of the co-crystallized ligand. And you can see here that we have a range of different ligands. We have peptide-likes, we have antiviral like the pociprevir drug, we have fragments, small fragments, larger fragments, small molecule inhibitors. We have also natural compounds like picoline, which is a flavonoid. And the idea was to study the specific interaction between each of these ligands and the enzyme. And we counted the number and the type of interaction for each ligand with the same enzyme and tabulated them. For example, histidine number 41. These are the type of interaction that participated in for each of these ligands. We have HY, it means a hydrophobic interaction, hydrogen bonding interaction. This one is a bi-bi interaction. We have the C stands for a covalent interaction. So for example, histidine 41, the overall count of the interaction with these ligands was a 22. So based on this count, this is just a summary of the bigger table actually. All other amino acids were counted and included in the study. Based on this count, the amino acids in the binding site were classified into one of three classes. The first class includes residues with very high count number, greater than or equal to 14. And they were the histidine. Now we will shortly come across them. The class 2 includes residues with intermediate count number, greater than 7 and less than 14. And the class 3, sorry, was the low count number, and they were somehow neglected because they were not very influential in binding these ligands. So this is the mapping of the key amino acids. The class 1 amino acids, which have very high count, in other words, they are involved or establishing good interaction with high number of co-crystallized ligands, were identified as key interacting residues, which are the methionine 49, methionine 165, and the catalytic diet histidine 41, cysteine 155, glycine 143, and the glutamic acid 166. The other two, histidine 164 and serine 144, they actually belongs to class 2, which are the intermediate count proteins. So now we have in hand the key interacting amino acids, based on which we will generate 3D pharmacophores. So to generate these pharmacophores, actually, we selected the crystal complexes. We selected the crystal complexes in which the co-crystallized ligand were establishing the highest number of interaction, and we ended up with 7 different crystals. And here we have the co-crystallized ligands. We have peptidomimetics, we have pociprevir, we have small molecules, we have picoline, which is the flavonoid. We have also a fragment, small fragments. Those complexes were studied or used to generate pharmacophores using a protocol available in Discovery Studio. And Discovery Studio, it is a modeling suite that has different functionalities. You can do molecular modeling, you can do molecular dynamics, pharmacophore modeling, ligand-based, structure-based design, docking, whatsoever. There's a lot of options available there. So I'm using Discovery Studio. And particularly in this case, we used ligand receptor pharmacophore generation protocol for all of these 7 complexes to end up with this different number of pharmacophores for each complex. So collectively, we ended up with 37 different pharmacophores. But when we studied these pharmacophores and we overlaid them all together, it was evident that only 3 of them, or those 3 of them, can account for all the 37 pharmacophores because they have exactly the same features. So we have pharmacophore 1 from this crystal structure, 6Y2F, and pharmacophore 2, plus pharmacophore number 1 from another crystal structure. And when you overlay all of them, you can see that they have identical pharmacophoric features. But if we look at this pharmacophore, we have 1, 2, 3, 4, 5, 6, 7 features. And the interfeature distance is quite small. And in this case, if you would like to use it as a search query, the number and the quality of the retrieved hits will not be that good, actually. So we edited that pharmacophore in such a way that we maintained all the features, pharmacophoric features that are accounting or representing the key interacting residues. So at first, we removed those 2 hydrogen bond donors to end up with a 5-feature pharmacophore. Now it's called pharmacophore A. Then we removed this hydrogen bond donor that is matching the carboxylate of the glutamic acid 166 to have pharmacophore 2 or B with 4 features. And the last pharmacophore was obtained by replacing this hydrogen bond donor with a negative ionizable feature. So now, if we would like to use these pharmacophore in virtual screening, any ligand that can fit any of them will be a perfect complementary ligand to the amino acids in the binding site of the protein. So we use the 3 of them, pharmacophore A, B, and C, in virtual screening of commercial databases. Then, as I just said, all retrieved hits were filtered. And this is a summary of the virtual screening process. So the 3 pharmacophores with the 3 different databases ended up with about 19,000 retrieved hits. After filtration, removal of duplicates and the bad binders that do not comply with Lipinski and Vipper rules, we ended up with 1,300 compounds. So these 1,300 compounds were docked into the binding site of the many protease, and their binding energy was calculated. And based on this docking and energy score, we selected only 28 compounds that have favorable binding energy scores. And this is a sample of the compounds that we obtained. This is the total binding energy. We can see that all of them, they have negative values. This is the virtual positive control, Posiprevir, which is co-crystallized with the many protease. This is the BDB code for this complex, 6WMB. And so these 28 compounds were really good in terms of the binding orientation and the scores in terms of docking and binding energy. So in order to shortlist even those 28, we performed ADMET descriptors calculation. We studied the pharmacokinetics and the toxicity. And all of these calculations, or all of these results, are based on calculations. So in real case or real life scenarios, it is highly recommended to take a lot of care towards the pharmacokinetic and toxicity properties of the lead compounds. And we just said that, or so that the drug discovery process is a very lengthy, very costly process. So it is much better to filter out any drug candidates or any lead compounds that can fail along the way because of its bad pharmacokinetic and toxicity properties. That's why we calculated these properties. And the ones that we calculated were the aqueous solubility, blood-brain barrier penetration, inhibition of the cytochrome P452-2D6, hepatotoxicity, human intestinal absorption, and plasma protein binding. Based on these calculations, 8 of the compounds showed very, very good results. So those compounds were actually considered as potential main protease inhibitors. And to make sure that those 9 compounds are quite novel, exact search was conducted using the SciFinder database, and none of them has been reported previously to be targeting any main proteases in either in SARS-CoV-1 or the SARS-CoV-2. And among those 9 compounds, in particular, I was quite interested in compound number 8. So let us have a closer look at the dot pose of that compound. We can see that the compound is perfectly fitting the binding site of the enzyme. It is occupying the 4 sub-buckets, the S1, S1 prime, S2, and S4. And also here we have a 2D interaction map. This is the compound number 8. And these are the amino acids, and these are representations. The dotted lines are representation of the type of interaction. And the good part that all of the 9 retrieved compounds, all of them, they are establishing a numerous interaction with the identified key interacting residues inside the active site of the enzyme. And here, for example, we have the methionine 165, we have glycine 145, we have the glutamic acid 166, and so on and so forth. And since this compound is quite interesting, why? Because it has been reported in the literature. It is co-crystallized with the X-linked inhibitor of apoptosis protein, the XIAP, with a BDB code 3CLX. And just to remind you that we use the 3 different databases. One of them is Maybridge, and the other one is the protein data bank, co-crystallized ligands. And the other one, or the third one, is drug-like diverse, which is a built-in database in Discovery Studio. Collectively, those 3 databases, they have or harbor more than 64,000 compounds. So out of these 46,000 compounds, we ended up with those 9 compounds as potential inhibitors. So the other good thing about this compound, it has a lot of known experimental data. It is a very potent binder to this particular target, and also it shows very good experimental ADMET properties. So unfortunately, it was difficult to run the experimental studies for these compounds. But as I said, we are talking about interdisciplinary collaboration in scientific research. So the idea behind this work, in fact, was to narrow the searchable chemical space for drug-like compounds, and this will this hopefully will help in identifying a new therapeutics for the COVID-19. So it was aiming to shortlist these compounds for other researchers around the globe who have the facilities, for example, the equipped labs that can conduct viral research. And hopefully, if any of these compounds get tested, there will be promising results getting out of it. And because we are, myself, I was interested in that one, MD simulation was conducted to further study the binding interaction. So you can see here, unfortunately, I'm not sure if the image is quite clear or not, but this is the binding site, and here is compound 8. And now it will be much clearer. So this rotation, just to have an idea about the protein, where the binding site, where the compound is being docked. And now we can see how the protein and the ligand are evolving. We can see that the protein, the ligand is changing the orientation a little bit at the very beginning of the simulation and maintain the same orientation throughout the simulation. And this was quite interesting because it means that this protein seems to be good binder for that, but it is not always good to judge based on your visual inspection. You need to calculate some values, and these values that I will come across shortly are very informative in this regard. So The values that I'm referring to are the root mean square deviation and the root mean square fluctuation. The RMSD, simply it is the standard deviation of the generated frames along the way compared to a reference point. And this reference point in our case was the first frame, the minimized frame. So it was studying how these conformations are changing relative to this one, and this is what we got. The other plot is the root mean square fluctuation. It measures the fluctuation of each amino acid or each amino acid residue of the protein throughout the simulation. In other words, it tells us which part of the protein was more flexible than the other. And in order to have an idea about whether our compound could be a good binder or not, another simulation for the apo form of the enzyme, which is the unliganded form, was run for the same period of time. And then the average RMSD and the average RMS values were calculated. Interestingly, in both cases, the values for the virtual complex were lower than that for the apo form. And this can be translated into stabilizing effect. So compound 8 actually seems to be stabilizing the protein. And if there is a good stabilization, then there is good binding affinity. So this compound, hopefully, if there's any chance to be tested experimentally, it might show really interesting results. And here we have, this is my final slide. And just to have a closer look at the different orientations. So this is at 0 nanoseconds, at time frame 0. This is the orientation of the docked ligand, compound 8. And this is after 10 nanoseconds of simulation. We can see that it is rotated roughly around 45 degrees and maintained the same orientation, more or less, throughout the simulation. Again, it is occupying the basic sub-buckets of the protein. Here we have a 2D interaction map of the complex, excuse me. Here we have a 2D interaction map for the same complex at 0 nanosecond and at 30 nanoseconds. In both cases, we can see that the type of interaction with the binding site amino acids is somehow different. But interestingly, in both cases, the key interacting residues were still in place. They are involved in essential binding interactions. So this leads us to very short concluding remarks that interdisciplinary collaboration is very critical, very crucial, especially in these circumstances. The global effect of the pandemic. Unfortunately, it affected the life of every single person in the world. It hit the four corners of the globe. So global collaboration is a must in order if we would like to fight this. And hopefully this work will be a very tiny contribution in this regard. The other thing that we can conclude that is COVID-19 main protease. Currently, it is attracting very much attention as a potential target for treating this disease. Also, computer-aided drug design has been proved successful in enriching drug discovery process in terms of time and revenue. And the structure-based drug design in particular is very indispensable, especially if we have a 3D structure of the target of interest. For those who are interested in this work and would like to know a little bit more, they can find more details because this work has been recently published in Journal of Biomolecular Structure and Dynamics. And at the end, I would like to thank the Faculty of Pharmacy at Jordan University of Science and Technology and the Deanship of Scientific Research for providing the infrastructure and facilities for this work. And here at the right-hand side of the screen, you can see where I live. This is Jordan University of Science and Technology. This is the Hospital University. And all of you, you are welcomed anytime to visit Jordan, to visit JUST, and definitely you will find a friend welcoming you there, which is myself. So you are more than welcome. And of course, at the end, I would like to thank every single one of you for your kind attention. Thank you so much.
Professor Nizar, for a very informative talk. Sir, on behalf of the participants, we got few questions. Can I narrate those questions?
Yes, of course, sure. You're welcome.
First question is, machine learning is emerging technique. Do you think that it makes a difference from the currently available computer-aided drug design approach?
Pardon me, would you please a little bit
Okay.
speak slower? Yeah.
No issue, sir. Machine learning is emerging technique. Do you think that it makes a difference from currently available computer-aided drug design approach?
Okay. It is very, very interesting, very good question. Actually, this is currently taking place in computer-aided drug design. It is not displacing computer-aided drug design. It is advancing the whole process. Now, I didn't talk in much details about the very specific issues of this technique. One of them, when I was talking about the docking and the scoring, we have different types of scoring function. And up to now, the bottleneck of any project related to structure-based drug design is the scoring function. You need to find a good compromise between the accuracy and the speed. And roughly, you can classify the scoring function into three major classes: the force field-based, and the empirical scoring function, and the knowledge-based scoring function. Recently, there is a fourth class, which is answering your question, which is the machine learning scoring functions. And they are very accurate, and I think they will have a very, very promising future. And it is not only about the machine learning. You could definitely know machine learning is like the random forest and other techniques. There's a lot of artificial intelligence is integrated in the process. So with the immense advancements in the algorithms, the computer power, I guess in the very near future, the AI and the machine learning technologies will boost drug discovery and other research areas in other disciplines of science. So definitely, yes, it will be very interesting to see that taking place in the near future. Yep.
Okay, thank you, sir. The second question is, how this computer-aided drug design is useful in tackling the future pandemic like COVID-19?
Useful in tackling, sorry?
Yes. How this computer-aided drug design is useful in preparing the future for the upcoming or whatever, the uncertain pandemic like COVID-19?
So the idea about using the computer-aided drug design, it is the enrichment. At the end of the day, you need to test your compounds in the lab. You can't rely solely on the scores because there's a lot of approximation. Even though the calculations are based on approximation, but they are really giving very good results. So the whole idea is about enrichment. So if, for example, you would like, or it normally takes, let us say, 10 years to find a cure for COVID-19. If you implement different techniques, including computer-aided drug design, those 9 years might be ending up 7 years. So you are enriching the whole process. And this is the core value of implementing computer-aided drug design in the drug discovery processes. Also, there's a lot of simulation, apart from drug design, that can use computer algorithms in pharmacoinformatics to predict the spread of the disease, for the pandemic issues, for the pharmacokinetic issues, there's a lot of computer implementation in drug discovery, in healthcare issues. And I think this will be a very integral part. And based on what Dr. Aaron just said, that the new technologies that are emerging related to the healthcare systems throughout the world, they are implementing a lot of computer technology within. So basically, these techniques and the advancement of these techniques will, I think it will somehow change the way we think, the way we do our research in the near future. Because if you go back in time, 20 or 30 years ago, there was very, very minimal implementation of computer sciences into drug discovery. Yes.
Thank you, sir. The next question is, sir, how to predict target for mutating virus like enzyme protease for COVID-19 virus?
How can you predict the importance of the protein?
Mutating enzyme.
Okay. So actually, the answer of this question is the essence of the interdisciplinary collaboration. Any drug discovery project in the whole world. It starts with a disease. And once the disease is known, you need to identify druggable targets, and not any protein in the virus or in a pathogen or is a druggable target. A druggable target, it means this drug can be, the function of that protein can be manipulated by small molecules. So in fact, this work is not my part. My part in computer-aided drug design is usually implemented in lead identification, hit identification, optimization, and in very, very small interface with validating the druggability of the identified targets. So basically, biomolecular studies, the biochemist and the biologist and the molecular biologist, they are the ones who identify that this specific target could be a druggable target based, for example, on mutation studies, knockout studies, and this kind of experimental procedures. But the implementation of the computer-aided drug design in this particular aspect is interfacing. I will not say it is a major rule. It is a very, very small rule. But from that step onward, it will be very important. Yes.
Okay, thank you, sir. The next question is, sir, in COVID-19 era, scientists has published, number of articles, including review articles and the research paper in various reputed journals. So the question is, how many pharmaceutical industries are, following these research or review articles to come up with or to combat this COVID-19 pandemic?
Okay, so this is very interesting, very excellent question. And to be frank with you, lately there was some concerns about the quantity and the quality of the published work pertinent to COVID-19. There's thousands of publications around the world talking about COVID-19, and some of the scholars are questioning the credibility of the results. So this is a very valid question. But if you go back a couple of months ago, and we would like to ask ourselves, when the breakout of COVID-19 took place, it was 9 months ago. And during that time, it is very, very unlikely for any mega pharmaceutical company to start, let us say, doing preclinical trials on treatment, not vaccines. Even the vaccination, which has a different approach, up to now, none of these pharmaceutical companies, apart from the Russian company, that are capable of producing a safe, effective vaccine. The process with the drug discovery, it is somehow more difficult. The very, very first slide of my talk was highlighting the duration of any drug discovery process, which takes roughly from 10 to 15 years. So it is very unlikely to have any drug candidate that will be surfacing in the near future based on this research. There's a lot of experimental work need to be done. A lot of preclinical safety issues need to be done. And a lot of preclinical trials need to be done before reaching the stage where we can try any of these compounds in humans. So, but the bottom line, this research definitely in other academic institutions, other research groups, and other pharmaceutical companies, they are sharing data, they are exploring the literature to identify potential hits that can be worthy of further optimization, that could be worthy of further testing. And hopefully the ones that I was talking about, these compounds that have been shortlisted, that could fall in the hands of some of the interested researchers that has the capability of doing experimental work. So what is happening here is integration of interdisciplinary research. So we need to wait a little bit more time, not a little bit more time, to find any good results for all of these research. Yes.
Okay, thank you, sir. Sir, as you know, most of the polyphenols give very good, document score for almost all targets, including COVID-19. What is your comment, the way forward?
Okay, so you can simply, I would like to say it this way, but hopefully it will be good to hear. I remember my supervisor was saying, if you are using a computer to run an experiment and a simulation, if you give a crap in, you will get a crap out. So usually you will always have data, but the idea is, how can you scrutinize this data? the preparation is very, very critical. So in my case, for example, you can perform docking studies without preparing the crystal. And in that case, definitely you will have some kind of biased or unrealistic results, because any crystal structure, we know there is a crystal, a crystallization artifacts resulting, for example, from a crystal packing. So you need to relax the protein a little bit. You need to solvate it. You need to neutralize it. You need to relax it before doing the docking. Also, you need to use different scoring functions. I just shortly mentioned briefly the different types of scoring function. You have so many different scoring functions, and the methodology in each of these scoring function is different. And based on that, the output will be different. So for example, for the same complex, the same ligand, one scoring function will give you a score of 5. The other scoring function will give you a score of 135 kilocal per mole. And also the nature of the interaction is very important. Some kinds of scoring function, for example, the force field based scoring function, they are somehow exaggerating the electrostatics. So if you are looking compounds that have ionizable groups with other compounds that don't have ionizable groups, that you need to expect to see some higher values for the ionizable ones, because the force field will calculate the electrostatics and it will give it a higher weight. Other scoring function, they will be basically interested or focusing on hydrophobic interaction and hydrogen bonding. So if your compound or the binding site is basically or mainly hydrophobic, or the key interaction are hydrophobic interactions, and your ligands are hydrophobic, then it is much better for you to use that specific scoring function. And because of these drawbacks and limitations, it is a common practice for all those who are working in the modeling field to do rescoring and consensus scoring, so that you can get rid of any bias that can result from using single scoring function. Besides that, you can do even more extensive calculation, like the one I just highlighted, which is using the implicit solvation. You can calculate the free binding energy. But this is actually more computationally expensive, and you can do it for, let us say, for a short list of compounds, unless you have very good resources, computational resources. So the free binding energy will account for conformational entropy, for the solvent effect, and by that you will have a more trustworthy score. And at the end of the day, if you are really interested in pursuing this complex up to the end, you need to run MD simulation. And based on that, you can calculate the free binding energy. And it is very, compared to the other scores, it is very, very accurate. So hopefully this has had answered your question.
Thank you, sir. Sir, we have a last question from my faculty colleague, Dr. Abhijit. Dr. Abhijit, please.
Hello, Professor. It was very nice presentation. I have enjoyed
Thank
it.
you. Thank you.
Two questions just to check. I have I heard that you screened 48,000 compounds to come up with this best 21 compounds. So how did you selected this database of 48,000 compounds?
How can I what? Sorry.
How did you selected the database of this 48,000 compounds?
Okay. It's really very interesting to listen to these questions. They are very specific and direct to the point. The chemical space, actually, it is 1 to the power of 60, which is more than the whole atoms of the galaxy. Sorry, not the atoms, all the galaxies, all the stars in the galaxies. So it is a very massive number. Now, you have different databases. You have zinc, for example, you have Kimball, you have BDB, you have Maybridge, you have Aldrich, you have NCI. There's a wide range of different 3D databases that can be used in virtual screening. And it is, at the end of the day, it is up to you as a researcher, based on your experience, on which of these databases that you will be focusing on. Myself, I have good experience with Maybridge. I have good experience with Aldrich. And why is that? Because they have a drug-like compounds. And they are commercially available. So if you'd like to test them, you can buy them straight away. And they are quite diverse. And previously, based on my little experience, I could manage to find very potent compounds based on these two particular databases. The larger databases, for example, the zinc or the other ones, there's a lot of available databases. You have millions there. And if you have good, really very good computational resources, you can download these databases and you can generate a 3D databases. By the way, if you don't have good resources, this is a process. Generating a searchable 3D databases is very computationally expensive. So there is no yes or wrong or no, sorry, let us say true or false, yes or no answer for your question, but it is a very critical question that searching the chemical space is very critical. And by the way, you can find in the literature that different researchers have screened zinc, for example, thousands of other compounds using different methodologies. So it is about how can you articulate the idea in your mind and whether this idea will lead to something fruitful or not. It is not only competing for publication. It is at the end of the day, it is science. If you are not doing good science, no one will listen to you. No one will buy your science. So you need to be smart and you need to be genuine and authentic. And at the very beginning of my talk, I said that the motive, the real motive of my work was based on moral issues, that I'm a drug designer and the whole world is suffering, and myself, my family, from this COVID pandemic. So I need to do something. And you never know. Maybe this work will be dropped away and no one in the whole world will have a look at it. And maybe some good or big pharmaceutical company, some of their researcher will say, why not to try these compounds? There are just a few of them. And to end up with having a lead, and that lead will be developed into a drug at some point. So you just do your part to the best of your knowledge and with the best of intentions, and leave it to the others. So so yeah, to make the answer, I know I'm talking a lot, so forgive me for that, but you have different databases, so many different databases. You can use whatever you want. If you have special interest or experience in any of them, you can go for that.
Actually, I'm working on natural product research, and then I was like curious to know that have you considered any of like the dictionary of natural products or anything related to that? That okay. So that I understand like some of the we have here Schrodinger software, and some of the studies like MD simulation that you have shown take a lot of time. It depends on your machine also. So for like for this 21 simulations that you have conducted, approximately what was the time duration that you it took to complete this experimentation?
So you are talking specifically about the MD simulation part?
Yes, yes.
Okay. So in fact, this simulation was run on a GPU cluster, and the 30 nanoseconds, it took, I think, around 2 days to finish. Because that was enough for me, because I just wanted to study the stability and the type of interaction and the behavior, the dynamical behavior. So you can run it as long as you want. One important thing, if you are interested or any of the attendees are interested in MD simulation, it is very important to ask yourself what I am looking for, because there's a lot of things at the molecular level that take place at different time intervals. So if you are interested, for example, with vibrational motion, a few nanoseconds will be more than enough. If you are interested in loop motion, then you need to simulate for hundreds of nanoseconds. And if you are interested in studying domain motion, then you need to run for much extended time. But there is something strange in MD simulation. What you are really doing, you are trying to explore the energy landscape. Remember, we are calculating the potential energy. So when you are doing your simulation, like you are walking on a terrain, you are exploring the energy landscape. And maybe you will run the simulation, and metaphorically, while you are standing on a top of a hill. But my simulation, the random seed for the simulation started for for example, it was starting from the valley. So I will reach a more stable complex much faster than you. And I can see very interesting results very early in the simulation in the trajectory. So the whole process, it depends on the efficiency of the algorithm. I was using AMBER 12 and the processors or the machine you are running the simulation. And nowadays, I think GPUs are overtaking CPUs in terms of MD simulation. So it is really good to do this because it is very informative approach. Hopefully this answered your question.
Okay, yes, sure. Thank you so much, Professor. Nice talking to you. Thanks.
Once again, thank you, Professor Nizar. Thank you very much for the time and the wonderful talk. Now I request Dr. Rakesh Tekade to. Now I request Dr. Rakesh Tekade to please come and give a vote of thanks. Dr. Rakesh Tekade.
Okay, thank you.
What a release, sir. What a day, I must say. It was 2 o'clock we started and now it's more than 5 past. Time really flew. So first of all, my very, very big thank to Professor Anand Paradkar, Professor Nizar Alshari, Mr. Arun Mishra ji for accepting our invitation and becoming a part of this webinar, who have really spared their time from this very busy schedule they are following. I thank all of you for your constructive dialogue and discussion. And thank you very much for your contribution and time in knowledge building. I thank all the speakers for taking time in precisely, accurately answering the questions raised by the participant. I thank you all very, very much from the bottom of my heart. I request all the participants to please join me for giving the speakers a huge round of applause. Thank you very much. I humbly thank Professor Kiran Kalia, Director, NIPER-Ahmedabad, for assurances, support, and of course, guiding us through the process of organizing this webinar. I would also like to thank the participants for their patience in attending the webinar, of course, for their very constructive, useful comments, as well as asking very nice questions, which are very informative, that again give life to this webinar. Friends, on behalf of the organizing committee, I want to express that we have tried our best to host you the best, as well as to make maximum of your time. I hope you enjoyed the talk and made it most useful for yourself. So, huge round of applause for our participants as well. Thank you very much. Thank you for attending. I thank Dr. Dirajan for helping management preparation for this webinar. I thank Ashish for taking care of the logistics. Friends, we have broadcasted this webinar in 3 platforms. One is on YouTube, another is on Facebook, and we are live here on Zoom as well. So during this many Havish, Shreya, and Nimit really helped us in assimilating the questions that were asked by the participants across this platform. You did a splendid job, all three guys. Thank you very much. And last but not the least, I would like to thank everyone for your time. Have a good evening. Please do not forget to give any feedback you have for us. We are really up for improving ourselves. And have a good evening. Thank you very much once again. God bless you. Thank you. So with this, we are stopping here. Have a nice time.